All right, audio good. Well, welcome everybody. Good morning to the 47th annual Texas A&M Sheep and Goat Field Day. My name is Reed Redden, and I'm a sheep and goat specialist for Texas A&M AgriLife Extension, and also interim resident director here at the Texas A&M Research and Extension Center in San Angelo, Texas. So, welcome to the 2020 version of our field day. 2020 has been an odd year. We wish you were all here, and we had hundreds of people out here for our field day, but we're going to have to do this virtually and join us in. So we should be live on Zoom. We're also going live on Facebook, so you should be able to tune in to both of those. We've got an exciting lineup today and tomorrow for the field day and expo. This morning, we're going to be all doing field day things, and we're going to be broadcasting live from a different spots here where we do a lot of research and outreach and demonstration and testing services. So here this morning with Daryl White, there's been some changes in our administrative model here at the Research Center. Dr. John Walker has stepped down and back into the research role. You're going to see him later today and him talking about some of his research and outreach efforts. I've taken on a portion of that role, and Daryl White has as well. And so I do kind of the overarching research goals, and Daryl does most of the day-to-day. So Daryl, you want to introduce yourself here for a second?
Sure.
Tell us what you think about being on the job so far since March, right?
Yes, sir. Yes, sir. March 1st. So name's Daryl White. I'm the senior administrator here at the center. I basically oversee the day-to-day operations. So since I'm relatively new, a little about me, I've previously managed 90,000 acres in Laredo, Texas, and most recently was managing 15,000 beef cows in Florida. And one of the things I've learned about being at AgriLife is that even though these are university managed and owned properties, we face the same challenges that all other producers face. And so as a result, the researchers here understand those challenges very well. And I'm excited about today because we're going to get a chance to hear some practical opportunities to face those challenges. So I'm excited about today.
That's right. Our mission here is ranching solutions. And so we're trying to test ranching strategies and things that help the industry do solutions. So first off, we're going to have some genetics and parasites. So we all know that parasites are a big issue in the sheep and goat industry. And then we're going to talk a little bit about nutrition and the things that go along with nutrition. And then we're going to also talk about some wool quality and wool testing. We also have a livestock guardian dog program, and so we'll give an overview of the research and outreach things that we've got going on there, and then a kind of a menagerie of other things. So we've got 12 different 10-minute presentations. So we're going to give you an overview of all the things that we do here at the Research Center, and then hopefully we'll be able to give you a better idea of what happens. A little bit of housekeeping for the field day. We're live. If you have questions, put them in the chat. I don't know that we're going to get to time to answer questions, but we'll definitely get back to you via email or other methods if you've got questions. And so, again, looking forward to a fun set, a day and a half here going virtual for the Texas A&M Field Day and Expo. Now we're going to cut away and go to Jake Thorne. Jake is down at our working pens. Jake is an extension associate, but he's also working on his PhD program. And so his main focus area is genetics and parasites. And so he's going to talk about both of those. So hopefully our IT folks are ready and they're going to cut away to Jake. So Jake, if you can hear us down there, take it away.
Good morning. Good morning, everybody. Thanks, Daryl and Reed, for the introduction. As they said, my name's Jake Thorne, and I am an extension associate here for Texas A&M AgriLife, and also a PhD graduate student. And so this morning, I'm going to talk about a really exciting new project that we've got going on here. And then the second portion of my talk, we're actually going to talk about how information that can be learned from that project and others that are similar to it can be put into practical application. So we'll get right into it. Gastrointestinal nematodes, specifically Tomachus contortus or barber's pole worm, are a significant issue for sheep and goat producers in the state of Texas. And what makes matters worse is that there are a relatively few amount of products or anthelmintics that are available to us here in the US to treat them. Also, this worm is highly prolific, and the efficacy of those dewormers is diminishing fairly rapidly. And so all in all, it becomes a very significant problem. Now, before we get any further, I want to review something with you really quick. This is the parasite life cycle, and this will help make the rest of what I'm going to talk about make some more sense. So the mature Haemorrhagis contortus lives in the abomasum of the sheep or goat. And as I mentioned, they're really highly prolific. They can lay up to 10,000 eggs per day. And those eggs pass through the GI tract, and they're deposited out on the pasture in the feces. And then depending on weather conditions, typically when it's warm and humid, those eggs will hatch, and the parasite will start to feed on bacteria in the feces. And that's about a 10-day process where it goes from eggs hatching to the third larval stage. So that parasite actually makes some transformations during that time period. And then once it reaches this L3 or third larval stage, it actually leaves the feces and it crawls up on the grass. And that's where the sheep or goat ingest the grass, re-ingest the parasite, and the cycle starts all over. That larva travels to the abomasum, and then it starts to mature into a mature worm again. From the time it's ingested until it starts laying eggs on its own, that's about an 18-day period. Okay, so with the loss of efficacy of anthelmintics, something that we really encourage producers to put into practice is genetic selection for those animals that are actually resistant to parasites themselves. So in the abomasum or inside the animal, we know that some breeds and even some individuals within breeds are better at defending themselves against that parasitic infection. And they're able actually to kind of flush those parasites through their system before they become a problem. And the immune system of the animal is controlled by the genetic code. And so what we really want to focus on is selecting for those animals with the appropriate genetics that make them more resistant for this trait. So how do we do that? Well, typically we would capture fecal egg counts from animals that are out on pasture. So they'd be grazing, they'd pick up worms. We would actually take a fecal sample from them, count the eggs in that sample, and use that as an estimate for the amount of worms that they've got inside of their system. But some things that make that a little bit challenging. First of all, it's weather dependent. So conditions need to be appropriate for a parasite challenge, warm and wet like we talked about, but also like we have had recently, several 100-degree days in a row, there's actually low parasite survival out on the pasture. So it's really timing dependent from a weather standpoint. Also, we can't control for how many parasites the animal ingests. How do we compare sheep A versus B if sheep A is consuming twice as many parasites as B? So that really maybe confounds some of our results. So something that we've done here, and actually these lambs that you can see right here behind me, are on an artificial parasite challenge. And so this is a project that's in collaboration with West Virginia University. And what they did is they took some eggs from a Haemorrhagis population, they grew them out to that third larval stage in the lab, sent them to us, and we actually inoculated all of these lambs. These 4 plus about 100 others, we gave them a known amount of L3 larvae, static across the whole group. So we're really starting everybody on the same playing field. And I also should note that before, ahead of time, all these lambs were dewormed, and follow-up fecal egg counts showed that they were free of parasites at that time. So we gave them the parasites, and then on weekly intervals, we are going to track their response. And so we do that by collecting blood for determining packed cell volume. Packed cell volume is the percentage of the blood that is red blood cells, and a measurement of anemia. Anemia is obviously a side effect of a Haemorrhagis infection. And then we also are collecting fecal egg counts. And so we're actually 3 weeks in. Week 1, there wasn't a lot of change in the response of these animals. PCVs and fecal egg counts were clean, not much to measure at that point. Last week, which was week 2, again, fecal egg counts were still 0. And that makes sense because it takes 18 days from that L3 larva to be ingested to start laying eggs. Only at 14 days, no eggs. But the PCVs or packed cell volumes were starting to come down just a little bit. Those parasites were starting to feed on the blood of the animal. Now, just yesterday, we collected samples again, and we got some really interesting results. The PCVs, again, were down just a little bit, but we started to see some eggs in the fecal egg counts. They actually ranged from 0 to 4,000. So we're starting to see some variation. We're starting to find some animals that are potentially more resistant or more susceptible to these parasites. So that's something that's really interesting. Now, to take this just a little bit further, we actually have collected tissue and DNA on all these animals, and we've ran that on the ovine 50K SNP chip. We don't have enough time to really get into genomics and the ins and outs of that this morning, but in short, what we're going to do is we're going to compare the results of our study to DNA on these lambs and potentially find some genetic markers for parasite resistant traits. So again, I think that that's something that's really interesting and exciting going on here at the station. Now, all of this, I think when you think back, what becomes apparent is that there's a really important tool in this process, and that's the fecal egg count. And so here at the station, we've actually opened up this service to the industry. This summer, we opened a lab that allows producers, outside producers in the state of Texas or across the country, to submit samples to us, and we will run those samples primarily for genetic selection purposes. If you're wanting to know just if your sheep or goats have worms and what types of worms, that's diagnostic service, and you need to work with your veterinarian on that. But for genetic selection purposes, feel free to submit those to us. We'll run those samples and help you identify some differences in that trait. If you want more information about that, I really encourage you to visit our webpage, sanangelo.tamu.edu, the testing services tab. You'll find the Texas A&M Fecal Egg Counting Lab, and certainly go there. You'll find the submission form. You'll find some videos about how and when to collect, and obviously pricing and all those kind of fine details. So we think that that's really interesting, and I think that that's going to be a great service that's provided to the industry. All it takes on your part is just to collect a fecal sample, put it in a box, send it to us, and you're going to get some really interesting results. So with that, before we get any further, do we have any questions in the chat box? If we don't, I can talk to you just a little bit more about what we're trying to do here. So we're trying to simulate a post-weaning artificial challenge. We feel that post-weaning is probably the most appropriate time to collect fecal samples and get those measurements from these lambs, because as opposed to weaning, there might be some effect of the dam and some influence on how that lamb was raised, litter size, and so that makes it a little tough. Yearling age, and when those animals start to get into production, again, there's some confounding variables on how the animal is partitioning its energy and resources. So we feel like that post-weaning is really a critical time for collecting data and getting the most accurate results for making genetic comparisons. So hopefully that explains a little bit further. If there's no more questions, we can.
Do you have a question?
Oh, awesome.
So, can you spray the pasture for worms to break the cycle?
Honestly, you potentially could, but you'd have to have a really small paddock in pasture. The question was, can you spray the pasture to control Haemococcus or worms that are out there? I think that would probably not be a really efficient process. Again, if you had a really small area, maybe that's an option. But when we start talking about larger acreage, that's really going to be a huge financial investment and probably really not feasible, honestly. Okay. Thank you. Let's go ahead and move on. So the information that is learned from this type of project, those fecal egg counts, that can be submitted to the National Sheep Improvement Program to generate estimated breeding values. And so estimated breeding values are a form of quantitative genetics, and they allow us to compare animals for just the genetic merit that they have. So if you back up to your most recent biology class, last year, 5 years, several years ago, you'll remember that the phenotype, in this case, the fecal egg count, is controlled by the genotype and the environment that that animal was raised in. And so what this program does is it really tries to limit the environmental effect out of that equation so that we can make a fair comparison on just the genetic merit of those animals. And what it does is it takes information on that animal's own performance, but also the performance of all of its offspring, all of its previous relatives, and compiles it into this one single figure, an estimated breeding value, or an EBV, and that really reports on the genetic value for that specific trait. So I've got a poster here that I want to show you. So this is a typical EBV profile. This is actually a bit of a condensed version. But these are 4 stud bucks that we have here at the station. And for these 4 traits, weaning weight, post-weaning worm egg count percentage, and number of lambs, wean percentage, you'll see that there's an EBV value in each one of these cells. And we're gonna just focus on this column right here. This is the only measurement here that is discussing parasite resistance. And you'll notice that there's figures both below 0 and above 0. And for all intents and purposes, I want you to think of 0 as breed average. And so this figure is a comparison against the breed average. And it represents, in this case, the percentage of parasites that this animal's offspring should have compared to the breed average. So we actually want a negative number in this case. So 175, ram 175, he's a negative 54. That means his lambs should have 54% less parasites than the average of the Dorper breed. That's a good thing. Conversely, these guys are all positive. They'll likely have more parasite, or their offspring will have more parasites than the average of the breed. Now, it's important to remember that these are estimates. They're not always exact, but they become more accurate as more records get put into the system, more progeny starts to get compiled into that EBV. So what does this mean? If you go to buy a ram or you go to a sale and you're presented with this information, you know, what can you expect when you breed one of these rams? And I want to show you something that's really neat. So those 4 rams were turned out in the fall of 18 for spring lambs in 2019. And what I've shown, or what I got here on this graph, is the average fecal egg count of their sire groups at a post-weaning time period, okay? And you'll see, to no surprise, that ram 175, his lambs averaged 970 fecal egg count, eggs per gram, at that post-weaning time period. Now, 331, he was positive for that egg count EBV. 12821 was positive for that egg count EBV. And look how much higher they are compared to 175. That really falls in line with that breeding value that was I just shown a second ago. Now, this orange line here, this represents the number of lambs that each of those rams had in that sire group. And that's on this far axis here. And so what you'll see is that 2506, this might be a little bit confusing. 2506 has the second fewest eggs per gram, his sire group, but he was a positive 63 fecal egg count EBV just a second ago. What's going on? Well, we really pride ourselves here at the station on showing you actual true data. This is not cooked in any way. So these are the true figures. And to make it more, probably a little bit smoother, we could have taken this guy out. But I wanted to show you this. First of all, what may be going on is that this ram only had 5 lambs in that sire group. So that's really not a large enough sample size to learn too much about that. But it does not necessarily reflect his EBV. But this is just one sample, one raw data set. So again, these rams were actually turned out for fall lambs that same year, last year, and this is what really makes this interesting. Again, we captured this information. Post-weaning fecal egg count by sire group, that's shown by the blue bars here. Orange line represents the number of lambs that they had at that time period. Again, 175, that negative fecal egg count EBV, 712 eggs per gram, his lambs average. That's really good. 331, he was pretty intermediate. He was just a positive 10 for that fecal egg count EBV. But look at 2506. That's the buck that had the second lowest egg count average that previous time period. Look at his lambs this time, almost 2,500. So that's almost double the next closest ram. That's where that positive 63 EBV really becomes apparent. We start to look at him and say, hey, you know what? We need more data. We need more beyond just those 5 lambs from that one time period to really capture his true genetic value for this trait. It becomes more visible, I think, in this data set. So that's what makes this information really powerful. These rams behind us here, all of those rams are in this group, these Dorper bucks. And the beauty of it is, if I ask you which ones are they, I realize that you're on a Zoom video here, but you couldn't stand here and pick them out just looking at them. Parasite resistance is a trait that we have to measure before we can really get an understanding of how an animal is going to respond to that type of challenge. We can evaluate structure and balance and muscle and masculinity and a lot of things visually, but parasite resistance, one of those traits that really can affect you economically, is something that we have to measure. Now, maybe you're a seed stock producer and you're going to collect data on your own flock, and this is really important to you, but maybe you're a commercial guy and you say, you know what, we're not going to collect data on our flock. We are just going to purchase animals that have these traits. And I think that's what makes understanding these EBVs really important. If you go to a sale And you see this profile for a number of traits, but specifically parasite resistance. That really is power. Knowledge is power in this case. And it allows you to buy those animals that potentially inject that genetic value into your flock. So I encourage you to check out the Texas Performance Sale. Today, we actually have an online sale that's open from 7 in the morning till 8 p.m. This evening. It's usually the sale that is in conjunction with the Texas Sheep and Goat Expo on Saturday, but we've moved it all online, and there's 25 lots in there, and I really encourage you to check that out online at Integrity Livestock Sales. You'll see that a lot of the animals that are displayed there really vary for that fecal egg count EBV. And now that you understand what maybe that genetic value can provide to you, if you purchase that ram and breed them to your ewes and what to expect from its progeny, I think that becomes really important information. One thing to note, EBVs are within breed comparisons. And so like in this Texas Performance Sale, we have Rambouillets, Katahdins, and Dorpers, and they all are going to have EBVs, or at least some animals in each of those breeds are going to have EBVs for parasite resistance. But you need to think of that comparison as just within breed and not across breed. So we hope this cleared some things up. Again, I think we've got a minute or two here. If we have any questions, I'd be happy to answer any. If not, we can go ahead and keep rolling along. If you've got a question, feel free to put it in the chat box there, and we'll go ahead and do our best to answer that. You know, this is a new format for us. We're trying to work through things, and so hopefully this still gives you an opportunity to interact with us and for us to supply some information back to you. If you've got one, feel free to ask it. If not, I think that I'll pass it over to Dr. Redden here, who's going to talk some more about estimated breeding values on goats for a different trait.
Well, thanks, Jake. Yeah, I think this is really interesting stuff. And as we think long-term about parasites, you know, there's not any new anthelmintics or dewormers that are coming down the road, and parasites are getting more and more resistant to them. So we need to be as proactive as possible about developing this parasite resistant genetics so we're less reliant on those pharmaceuticals in the future. And so, you know, our seed stock producers really need to commit to it. And the best way is our commercial people is just demand breeding values, particularly for parasite resistance going into the future. So I'm going to transition a little bit. We're going to stay on the theme of estimated breeding values, and we're going to move over and talk about some juniper eating goats. So bear with us here as we move. All right, Corey is doing a fantastic job videoing this. We're going to have her come right over here, try to block the wind noise a little bit, get pictures of some goats. All right. So when we think about genetic selection, oftentimes we're really thinking about things like wool production or parasite resistance or growth or reproduction. We don't really think about some of the other opportunities with small ruminants, and that's from a land management standpoint. So we have some goats right here. These are actually what we originally called the super juniper eating goats. That we also go by the name of AgriLife cedar eaters, which we can use an acronym of the ACE goats, which is easier to say, so we like that. These goats have been bred for over 10 years to eat more cedar, or technically juniper, than a regular goat. And so the goat industry has been in Texas for a long time. One of the greatest values originally for goats was brush management. The older ranchers knew that if you had goats, they would manage brush. And so brush encroachment is a real problem on a lot of the lands in Texas because that brush will overtake the lands and we can't grow grass for livestock, and we just gotta get an imbalance here. And goats are a great tool to biologically control brush encroachment, particularly juniper. So juniper, or we commonly call it cedar, is an evergreen, so it stays green year-round, and goats will eat juniper. The average goat eats somewhere in the neighborhood of 25% of its diet. It kind of depends on the time of year, what other things are out there, and whether that goat will eat juniper or not. But since we knew that, scientists here at the research center, before I really came back to Texas from some of my graduate experiences and other professional positions throughout the country, had developed these goats. And so what they did is they took goats, put them in a pen, and fed them a known amount of juniper, and then collected feces from them and created a quantifiable test using near-infrared spectroscopy. Dr. John Walker will talk about NIR, that technology a little bit later today, for this purpose and also for wool quality assessment. And so they quantified the ability to predict how much cedar they're eating, because it's hard to know without going out there and following a goat around all day and watching how much they eat of something, what their amount of cedar intake is. So they've developed a test for it, and then selected a high line and a low line. So bred the high eating female and male goats to each other, and then bred the low juniper consuming males and females together, and created divergent selection. And so what they've actually created, geneticists here generated estimated breeding values. So the same thing that we use for parasite resistance, this is a known thing in the genetics world. And so these two goats right here is 6850 is the black goat, and then 6508 is the red goat. So these were a Spanish boar cross composite breed that's very common to Texas when they started genetically selecting them. And so they look like goats, like range goats. But if we walk down here a little bit, Corey, these are the low-line goats. So the first goats over there are expected to eat 15% more in their diet than the low-line goats. So there's a 30% difference between the two of them based off of an EBV. Now, I know a lot of our ranching community can be skeptical about what this is and how that was generated and what it means, and will it be predictable on my place. And so we actually run a Angora pasture performance test for Angora breeders, and we do a juniper intake in that pasture performance test. But we also run these high line and low line goats in it to show them what a goat bred for high juniper consumption will be in comparison to their animals. And so that's what I've got here. So here's the EBV that I talked about before for 6508 and 6850 is a positive 15, and 6595 and 6681 are these two goats. They're low, so they're eating 17 and 15. Now this is based off data from themselves, data from their mother, data from their siblings. Breeding values take a lot of background information to put that all in there. But these goats were actually run, not all of them, but most of them were run with the Angora pasture test, and we did an NIR on themselves and to predict juniper intake. So in 2019, 6508, which is the red goat that we showed you before, ate 33% cedar, and in 2020 he ate 31. Okay, and then his EBV is a positive 15. All right, so that's pretty good, eating 30%. Here's 6850. He was actually not on the 2019 test, but he was on the 2020. So just slightly less from 31 at 26, and that was his EBV. Now these were the goats that were bred for low intake, right? This goat, 6595, which is the goat closest to us there in the shot, I don't know if Corey's got that in there, only ate 6% cedar in its diet in 2019. Very repeatable in 2020, ate 4%. Okay, this 6681 ate 8% in 2019 and 18% in 2020. So what this is kind of showing you is there's some variability, but the technology is very accurate at predicting juniper intake. I'm not going to get into this. We're probably going to run out of time on it, but we'd love to answer questions, you know, either through the chat box now, or you can send us something and we'll get back to you later. But the take-home message is goats are a great biological tool to control brush, and we can actually breed goats that eat more brush than the others. So if you're gonna use goats in that biological control program, why not select those that are gonna eat twice as much brush as another? And we think about, okay, they're gonna eat brush, that maintains our land, but that also allows us to run more sheep or cows that are more grass eaters, because the goats are eating the trees and keeping the trees from encroaching on the land and opening up the landscape to grass and weeds and a number of these other things that we tend to think are more preferential. And so there's a lot of wonderful tools out there with technology, but we have to implement them. So my take-home message for this that I'm going to leave you with is the Research Center developed the technology. Okay, we've proven that it works, but we're at a stage where we no longer are doing this because we have to determine pedigree. These goats have to be born in a pen. We have to do all of the testing. It costs us a lot of money to keep this going, and it is time for this technology to be commercialized. And so if you're watching this today or you're watching the recording later and this really intrigues you, I highly encourage you to get into contact with us, because we need to get this technology in the hands of people, and then they can go amplify it and breed these goats for marketing purposes. And I don't know if you're going to develop a breed association with it, but we are very willing to work with you in any way, shape, or form to take the technology to the industry and let the industry run with it. So with that, I think I've got a little bit of time here. If we've got any questions coming in the chat box, we could entertain some of those. I think this is one of the technologies that shows the teamwork that we do here at Texas A&M, both from extension and research, but also across discipline. We have range ecology people that are in this because of the juniper encroachment and how that works. We have animal geneticists that are involved in it, and even kind of the nutrition side, because we're taking it from a fecal sample and how nutrition influences their intake. So agriculture, managing animals, managing rangelands is a very complex thing, and we try to bring all people that are very knowledgeable in each of the areas together as a team to develop technologies and ranching solutions for our landowners and sheep and goat raisers here in Texas. So Dr. Hale, do we have any questions coming in? We've got not Dr. Hale here, not seeing anything in the chat box. So again,
so is this a concept that could even work in sheep? I know they're totally different beasts, but is there something you can do in sheep?
That's a great question. Is this a concept that works in sheep? It would be, but it's sheep are not as much of a browser. So a browser is the term that we call goats are browsers, meaning they eat leaves, shoots, and fruits from woody plants. Sheep will eat somewhere in the neighborhood of maybe 10% or less browse in their diet, but they don't have either the biological, the anatomic ability that a goat does to stand on its back feet and reach up there and grab stuff, as nimble of a mouth. But the main thing is they don't have the liver capabilities to digest some of the things that are in cedar that prevents sheep, cows, a lot of other animals from eating it, that goats possess that ability. White-tailed deer have some of it, but goats probably even more so, especially our ace goats. Fantastic.
Have there been any studies about goats eating salt cedars?
There have. I'm a placeholder for this. This is not my area of expertise. You know, Dr. Walker and some of the others that were involved in this technology would be better at that. And so hopefully there's a name on that comment that comes in. We'll get back to them. I know goats will probably browse on some, but to what degree, I don't know. Good question.
So how does it affect the taste of the meat?
So there has been research done here on feeding juniper and how that affects the quality of the meat, and it actually did not have any negative attributes to it. Dr. Travis Whitney is our nutritionist, and he's developed a wood to feed program where they actually feed juniper at known amounts in a diet, harvested, dried, ground juniper, and with a little bit of juniper in his in the diet. And you'll have to get with him to get the data on it, but I think it actually improved eating experience, which is not what we thought. What we think is kind of the gaminess of deer, the gaminess of goat to some people is attributed to their diet. Yes, diet influences the eating quality, but eating more juniper does not appear to have big negative impacts on meat quality. Good questions.
So how many total goats have you bred?
So this project has been going on for well over 10 years. So it's in the background. There's around, I don't have the exact numbers, but it's around 200 to 300 goats that are either a high line or low line. And we're slowly liquidating the low line and just maintaining the high line. We're keeping some of the lows around, but not as many as there were before for research purposes.
And you're doing comparisons with Angora? I mean, do you see differences in Angora?
There was an initial project with Angoras, but they did not see the genetic diversity that there was in the meat goats that allowed them to make more progress genetic selection with the meat goat than the Angora. Odd thing is in 2019 in our Angora pasture performance test, the Angoras averaged somewhere around 40% juniper intake, which is fantastic. But then in 2020, different year, different pasture conditions, the Angoras only ate 10% juniper. So there's a lot of variability in the average across the group, where these goats consistently ate the same amount year in, year out. Good question. All right. I think we're at a break point here. So all of this is made possible by our generous sponsors. And so we're going to go into a 5-minute break. You can go get some more coffee or tea or water or whatever your beverage of choice is. I believe they're going to play a short video to recognize the sponsors of these field days and expos. They've been sponsors of us for many years. They've continued this year, so we really thank them for that. So keep an eye on the sponsor video, get yourself something to drink, and we'll be back in 5 to 7 minutes.
In 1948, William C. Young had a dream: to support agriculture and the hardworking people who feed the world. And for over 60 years, Ag Workers has steadily grown by holding true to principles of loyalty, fairness, and service. And today, Ag Workers has expanded by providing access to opportunities that enrich the lives of its membership.
Ag Workers is a lot like family, and you trust your family. I trust the employees, I trust the service.
People love Ag Workers because of the relationships that we have with those people.
People don't thank you for your insurance. Our people do. We work for them.
They truly do look out for what's best for their customers and try to provide the absolute best service that they can.
Built on a solid foundation of strength, trust, and security, Ag Workers is more than just a business. It's people taking care of people. Ag Workers Insurance, a legacy of trust since 1948.
Good afternoon. My name is Gerald Hobson, and I am with the Bear Range and Pasture Group. Want to wish everybody the best of luck during this virtual Texas Sheep and Goat Raisers Association meeting. And we have introduced 2 key products into the range and pasture markets in Texas this year. Angora is a new tool, new technology, new chemistry, new molecule that's going to help those that have problems with mosquito and weed setch manage those problems better than they ever have before. And then in the warm season perennial grass markets, we're currently introducing Resilon, which is going to help manage annual grasses and broadleaf weeds on your warm season perennial grasses. So if you need more information, contact me or Dr. Alan McGinty, who lives right there in the San Angelo area, or any of the others of our range and pasture team. We'll be glad to help you. Once again, good luck to you, and I hope you enjoy the meeting.
It's hard to let something that's been in your family for generations go, so it feels good to make a go of this. I'm Tyler Black. I'm a 4th generation farmer here in Bailey County, Texas. We grow corn, milo, wheat, raise cattle. Working with Capital Farm Credit has been great. I mean, you're not on your own when you go into Capital Farm Credit. They treat you more like family. One of the best things about Capital Farm Credit is they speak our language. Together we're much better.
The American Goat Federation represents the interest of every goat owner in the United States. AGF travels to Congress each year to present producers' concerns and takes part in USDA and other agency meetings throughout the year to make sure the goat industry has a voice in decisions that affect goat producers. In addition, the Federation provides accurate information to the general public about goats and goat products.
EcoDeer, Ecology, Economy, Results. Brought to you by Palmer Feed. Family owned and operated since 1943. For more information, visit our website, palmerfeed.com.
No one ever said sheep and goat ranching is easy. Day in and day out, it's a tough job. Part of our mandate at Lakeland Farm and Ranch Direct is actually listening to our customers and finding solutions that actually work for you and your farm. Whether it's handling, trimming, sorting, or crowding, Lakeland has the sheep and goat equipment to help make your day just a little bit easier. Our Deluxe Spin Trim Chute is a farm favorite for many sheep and goat farms that need an easy way to trim, tag, and vaccinate their animals. The working chute and crowding tub is a great system to help crowd your sheep and goat and make your operation an efficient one. The Lakeland Easy Clamp is the go-to product for high-volume sheep and goat handling, perfect for drenching, vaccinating, dagging, and more. Whether you need handling equipment, fencing panels, sorting attachments, or weigh scales, Lakeland can help build a custom sheep and goat system that works for you and your farm. Through our farm direct delivery, our easy-to-use premium products, or our exceptional customer service, Lakeland Farm and Ranch Direct is the only provider of sheep and goat products that make life easier for today's rancher and the hardships they face.
Hello everyone, my name is Wade Polk, and I'm the branch manager at Central Texas Farm Credit in San Angelo. We are proud sponsors of the 2020 Texas Sheep and Goat Expo, and we want to help you with your financing needs. We finance and refinance farm and ranch real estate loans, as well as providing loans for operating, equipment purchases, and livestock. We understand the ins and outs of agricultural financing, and we can customize a loan to fit your specific needs. To learn more, stop by and visit with us at 1021 South Cohenheim, or visit us online at ranchmoney.com, or call us at 325-658-8545. Thank you.
It's more than a piece of land. It's a place where you create memories and leave a legacy. It sounds like a dream, but at Capital Farm Credit, we'll help you make it a reality. As the most experienced rural lender in Texas and a cooperative, we're with you every step of the way. Your goals are our goals, because no matter what your dream might be, we're all in this together. And together, we're better.
Weeds will rob me of my investments, because any of the weeds, they'll take up the nitrogen and they take up the water that I need for the grass. We need the grass for the cows. The weeds are not palatable to cows. They will not eat them, or if they do eat them, some of them may be toxic. So there's a return on investment by allowing there to be more grass available to be grazed by the cattle. We have tried to manage our forage to provide those cows those protein and energy needs to be successful.
Okay. Are they? We ready? Okay, welcome back, everyone. We're here with Dr. Doug Tollison, and he's going to talk about quantifying the amount of forage that these cedar trees out there, because we just talked about the goats and getting a high-consuming goat to manage this browse. But it's a good idea to identify how much of this browse is out there, and if you want to manage it, to what percent and how many goats you would need to hit that. And so Doug's done some recent research on this, and so he's going to talk to you about quantifying the amount of cedar forage that's in your pasture. So, Doug, take it away.
Thank you, Reed. Again, I'm Dr. Doug Tollison, and I'm an AgriLife range scientist, and I'm located at our historic Sonora station, which is about 100 miles south of where we're standing here today. Again, my topic is going to be cedar or juniper, depending on how scientific you are. And we're going to talk about two different ways of being able to quantify the amount of material that you have out there on your land. Now, we did some research that's been made available to you, I believe, in a Rangelands publication. And we're going to talk about an old school and a high-tech method of being able to make this determination. Okay, so I'm going to start with the high-tech version first. And so this is a graphic of some juniper trees that we collected, harvested, counted all and measured all the different types of material that's in that each particular tree, and came up with a total biomass and then a biomass for different fractions of that tree. How much would be browsable, how much would just be stems and burnable, and that sort of thing. So you can see from this imagery here of two different size trees, small and large, small being about 3 feet tall, medium, instead of large, medium being about 3 to 6 feet. You can see the images on this side here. They're in a true color, laser, 3-dimensional imagery that was produced by LiDAR. LiDAR stands for light detection and ranging. And then on the other side, you can see the color ramp that's been applied to that imagery to identify what material would be available to a browsing animal. The green or blue would be available to animals that are pretty much any size, and it would be in a 4-point stance or a quadrupedal feeding stance. The yellow would be available to larger animals, or those who are assuming a 2-point stance or a bipedal. And you've seen that a lot with your goats in your pasture, standing up on 2 feet, accessing forage further up into the canopy. The red then would indicate that forage which was not available in these trees that are greater than 6 feet tall. Now, we can do that for individual trees, or utilizing some of the technology that we have available to us now with drones. We can put these on fixed wing or helicopters. We can even get this through satellite imagery. You can see how we can move from an individual tree to a landscape using this aerial type of technology. So our goal with this technology is to be able to, again, possibly through radar, through satellite imagery, or through fixed wing or drone type applications, be able to collect this imagery on a landscape, one of your pastures, let's say, determine the biomass that's out there, and then with some other sensors, we can even actually detect the chemistry. So we may be able to get a 3D image of your pasture and tell how much protein is out there as well as how much biomass. That's coming down the road with the research that we're doing. This is probably 5 or 10 years away. So what can we do tomorrow if we want to go out on our pasture? Well, armed with things that are as simple as measuring sticks to determine height or width of a tree, a tape measure, we can determine how much forage is available in these different size trees. So in that paper that you'll see, if you want to go read it, we did these measurements on different size trees. A tree of about this size is going to produce about a ½ pound of browsable dry matter forage. So basically the needles, and then up to about a No. 2 pencil size or about a 1/4 inch size stem. All of that would be available to browsing. About a ½ pound of dry matter in there. Tree of this size, an average, it's going to produce about 5 pounds of browsable forage. If you get up to a 10, 12-foot size tree, just considering that, which is below that 6-foot mark, which would be the browsable forage, that browse line that you'll see on some of your pastures, you can get about 25 or 30 pounds of dry matter forage out of a tree that size. So if we know what size tree we have, know how much forage that produces, we can then go out in our pasture and mark off some blocks, count the number of trees of these different size classes, scale it up to how many acres we have, come up with a total pounds of forage. So let's kind of walk through that exercise. So if I've got a pasture that has a distribution of trees in it, something like this, it's not uniform. We're going to need to go out and put about 3 different blocks that we'll count in each of these different forage densities. Okay, let's assume that we have a pasture that's relatively uniform. We'll walk through the math that way. So if I've got a 100-acre pasture that's, again, fairly uniform in distribution of juniper, I'm going to need to mark off some blocks that will give me the ability to count about 10 or 12 trees in the block that we mark off. For 1/8 of an acre, it's going to be 50 feet on each side. 1/4 of an acre will be 100 feet. 1/2 an acre will be 150 feet. If we do the 1/4 acre, obviously we're going to multiply by 4 to get pounds of forage per acre. And then we'll multiply that by the number of acres in the pasture. So if I've got a 100-acre pasture, I've got 10 trees in a 1/4 acre block, that's 40 trees. and then I can multiply that up. If I do that for a 100-acre pasture where I've got 10 medium-sized trees, 40 trees per acre, that's going to be about 20,000 pounds of dry matter forage. So if I've got a 75-pound goat who's eating about 3% of her body weight a day, that's about 2¼ pounds of dry matter. If I have 100 of those, that's going to be 225 pounds. So 100 of those goats eating that much forage a day on that pasture with this distribution of trees would provide about 90 days of forage if that were the only forage out there and if that's all they were eating. Now we know there's going to be other forage. There's going to be other types of browse, oaks, there's going to be forbs, there's going to be grasses. So if that diet was about 25% juniper, that distribution of juniper would feed those goats for about a year. So that's a pretty rough estimate, but it gives you an idea of how we can quantify the amount of juniper that we have out there, how we can walk through that math and determine how long that would feed a number of amount of goats. So I hope that what we've done here is giving you an illustration of some future technology that's coming down the pipe and give you something that you could take out with some very simple tools and go do by yourself or with your family tomorrow. So with that, I will close and we will answer any questions, and I hope you enjoy the rest of the workshop.
Do we have any questions coming in on the chat? All right, thanks, Doug.
Thank y'all.
I got distracted. Did you mention our extension publication that should be coming out?
I did not, but there is one that's going to take you through what we did with the research paper and put it in a little more practical terms. And that'll be authored by Dr. Triplett.
Right. So be on the lookout for that. Hopefully that'll come out this fall. So we'll have an easy how-to guide to be able to go through some of this technology and implement it on your own place. So thanks, Doug. Appreciate that. Great work. Now we're going to cut away and go to Dr. John Walker. Unless do we have any questions coming in on the chat box? No? Okay, we're going to go to Dr. John Walker, who is at one of our water reservoirs out in a pasture, and talk about some water monitoring technology. I know we're in the nutrition section. People often overlook water. It's the most important nutrient. An animal can will die the quickest with a deficiency of water than anything else. And so monitoring water in the pasture is very important. So hopefully we're ready to go, and we're going to cut away over to Dr. Walker.
Well, good morning. I'm John Walker, and thank you for attending our virtual field day. I'm going to talk today about the most important nutrient for livestock, for people, anybody, and that's water. People spend many hours every week just checking to be sure that that your livestock have water. They may not worry about anything else, but we know if they don't have water for 48 hours in this 100-degree weather, you're going to lose some livestock. And if you've ever experienced a water system failure like I have and actually lost stock, operate ranches that are 100 miles from your house, you know how important and how much effort it takes to just to be sure that your livestock have water. So I'm going to show you 3 systems today that are ways to monitor the level of water in your reservoirs. We're going to start over here. I'm going to start from the most sophisticated to maybe the least the cheapest. This system is IDT. It's a rural water company, and it's got several elements. And it's the most complex, but it also gives you the most information. It communicates with a satellite, so you can put it any place. You don't have to have cellular connection. The others do. This system also has a cutoff valve so that you can remotely shut your water off if you find a system failure, a leak, a broken float valve. So the base system costs $150. This cutoff valve is $750 for a total system cost of $1,800 and an annual subscription rate of $90. The next system we're going to talk about is the Aguacheck. Okay. And this system has a solar panel, so you never have to worry about batteries or anything. It's the simplest system that's out there. It runs $750 with an annual subscription of $240. does require a cellular connection. So if you don't have cell service, this won't work for you, but it's a very simple system. All you have to do is attach it to your reservoir, throw this pressure transducer in the water, and send it a text message, and basically you're good to go. And it'll send you a text message every day at whatever time you ask it to, letting you know the level of water in your reservoir. Final system that I want to talk about is a game camera. There's lots of these out there. I really like this model. We've used some other ones, but this is a fairly new one. What I like about it is it has an attached solar panel. You can set it up for time lapse. This camera it lists for $500, but you can often find them on sale for $300. If you need an external antenna, you can attach that to it to get better cell service. That's an $80 edition, and it has you can get 100 pictures a month. That'd be like 3 a day, and there's no subscription cost to that. You do need at least 2 and preferably 3 bars to make this work. So those are the 3 systems. And now, Blair, if you'd show my first slide, I'm going to show you what kind of information you get off these systems. Tell me when the slide comes up. Is it okay? So this first slide shows you the IDT information you get, and the thing about it now, this system is a little more complex, and you can get it on your smartphone. It's a website. It's not an app, but if you look on that table to the right, it shows on the far right column, it shows the level of the water in tenths of a foot, and the next column shows how much water has been used each day, how much has been metered out. Next slide.
We're gonna wait for just 1 second to make sure that we have the correct slide up here.
This is what I'm looking for.
Yes, sir.
So the slide that's coming up shows you the information you get off of the IDT system graphed out. And what I really like about this system is that water usage, flow, gallons per day is much more sensitive than the level in the reservoir. And so if you look at that, the blue line, look at that peak, which is how much water is being used. So we suddenly jumped about 3 times as much water in one day as we'd previously been using. But if you look at the orange line up above it, you don't see all that much change in the level, particularly if you have a float activated pump on your system. You're not going to see it. It's really useful. And of course, if you see that and you have this automatic shut off, you can shut this off and not lose a reservoir full of water while you go fix it. Okay, the next slide is our Aguacheck. It sends a text message to your phone. It's very simple. You set it up and you don't have to worry about it after that. Is that enough?
Yes.
When you set it up, you'll tell it what time you want to receive a message. I set mine to come in 8 o'clock every night, so I know when I go to bed if I'm going to have to go fix water in the morning. However, it also has options of if the level drops faster than normal, it'll send you a text message. This system checks the level of the water, wakes up once every hour to check the level. If it sees it's dropped more than normal, it will send you a text message. So you can see that at 9 o'clock in the morning, I got a text message that said that it had fallen 2% in the last hour. Okay, next slide is the spy point. You have to install an app on your phone to look at these pictures. And so is that up? Okay. And so this just shows you, this camera is set up to take 3 pictures a day. And as soon as it takes a picture, this is on, I have 3 different cameras on 3 different reservoirs, and it just shows you a day's worth of pictures on them. You can enlarge those pictures. And so if you'll show me the next slide, here's kind of a close-up of looking at the level. You can see not only how much water's in the reservoir, you can see that your pump's working. It's delivering water, okay? The other nice thing about it is if and we've had this happen, if something falls in the reservoir and dies, you know it, and you can fish it out, okay? So that's an advantage. Another advantage for a lot of people is it's visual. You know the system's working. It's not sending you a number and you're wondering, is that number right or wrong? You see the picture, you know what you got. A lot of people like this. Next slide, please. And so I installed one of these systems just about a week ago. And the really neat thing, in a sense, was that the rancher I did this for had a water system failure the next day. And so the first one on the left, you can see where the marker is at the top of the picture. That means that his tank has gone down really fast. The next one, that was at 7 in the morning. You can't see the marker anymore. On the next one to the right, it means it's still falling. He was able to go out there, shut his water off. What it did is saved him from losing a reservoir full of water and getting air in his line. Last slide, please. So this last slide just shows you the break-even cost of how many trips you can save over different distances. And so, long story short, is if you live 40 miles from your property, any of these systems will save you money. Well, after 10 trips, you will be saving money. So which one should you have? Okay, come back to me now.
Yes, sir.
Which one of these systems do you want? Well, first off, do you have cell service? The answer is no. IDT is the only one that's going to work for you. If it's yes, any of the others can work. Is ease of use your priority? If the answer is yes, I would take the Aguacheck. If it's no, and low cost is your priority, I would go with the game cameras. If low cost isn't your priority and you want precision and versatility, then I would go with the IDT system. Any questions?
No, sir, we don't have any.
Okay, very good. Well, thank you everybody for watching. And if you have questions later, you can contact me and I'll try to help you out.
All right, thank you, Dr. Walker. Interesting stuff. We're back here at the nutrition barn now. Dan Quatros is going to finish up this nutrition section here and talk about some research that they're doing and do kind of lead in and a little bit of a show and tell from a nutrition standpoint. So thanks, Dan, for being here and walking through our attendees on this nutrition and feeding trial session. So why don't you go ahead and take it away?
Thank you. Thank you, Reed. My name is Dan Quadros. I'm a postdoc research associate here in Texas A&M, AgriLife in San Angelo. I work for Dr. Travis Whitney in the Livestock Nutrition Program. And I'm here to talk about sheep and goat nutrition. And why sheep and goat nutrition? Why is nutrition so important in your production system? Well, you may be thinking, if I don't eat, I don't live, so I die. So you are right. So, but in this business, we're not only feed the animals to keep them alive, but we need to make money. So nutrition is very important for reproduction because it affects reproduction. And reproductive efficiency affects your profitability. So it's very important in your system. Also the animal health. If the animal don't eat well, if the animals don't eat well, they're gonna get sick easily and they're not gonna recover soon. So nutrition, in addition to that, nutrition is representing about 60 to 70% of the cost of production. So it's very important, your nutrition program. But to start in your good nutrition program, you start having feed, your balance between the supply of feed supply and feed demand. So you have to be efficient in producing feed. So this is the first point. Well, talking a little bit about the nutrients and how the feeds are classified. First, I think was very important this previous section about the water, the importance of the water as the most important nutrient, the availability of water, the quality of water. So it's very important, not only microbiology, but the chemical analysis of this water, the distance of water in the pasture. So everything about water is important, about half gallon to more than 1 gallon, depending on your if are lamb or a mature ewe. But about feed, come back to the feed here. First classification that you can talk is about roughage and concentrate. So let me show you here. So what is name roughage? What is name concentrate? So maybe the energy that is in this bale of hay is in this beaker of concentrate. Okay, could have a little bit of exaggeration here, but it's for education purpose. But when you have the roughage, when you have the forage, the fiber is high. If you remove all the water, we call dry matter. So the fiber is more than, the crude fiber is more than 18%. Actually, when you see the fiber, the carbohydrates, that is the structure of the plant, like cellulose, cellulose, is sometimes 60, 65% or more. So if I have the space, let's say like that, like 100% and 67% is fiber, where is the protein? Where is the energy? So because that the concentrate that's have low fiber, less than 18% of crude fiber, we have more energy in the form of starch, that's no carbohydrates in the form of the starch, and protein as well. So let's come back here to our table. This is a display that we have a little collection. So first we have the forage here. So the forage in your ranch, you can think about range plants. You can think about perennial or annual cool seasons or warm season grasses or legumes as well. So the forage, here we have the Bermuda grass, very common, alfalfa, Southern grass. So in my point of view, you have to be efficient in your range to produce your own roughage. To buy roughage, so nowadays the price is increasing of your product, but when you see a bale of hay, for example, increase 60%, you know, in this coronavirus, and you have drought as well. And the roughage over here, to complete my collection, is agricultural residues that you can use. So you have also to think about when you're gonna buy this roughage or your feed, and have this balance between feed demand and feed supply. So you have the wheat hay over here, corn stover, some agricultural residues, very low quality of corn cobs that we are researching here about corn cobs in the sheep diet. Cotton gin trash, that's a residue very common in the southern USA. Cottonseed hulls is very common as well. And ground juniper. So you saw Dr. Tollison talk about the cedar. So after almost 20 years researching about ground juniper here, Dr. Travis was pioneer in ground juniper. Finally, last year, we got approved juniper as a feed ingredient. So now we can use in livestock ration. So it's very interesting because it's a very competitive cost. In his research, he transformed a problem in a solution. It is very nice. Let's now switch to the concentrates. We divided the concentrates into big classes, energy and protein. So when it's less than 20% of protein, crude protein, is a lot of starch, a lot of energy there. So we call energy concentrate. So the most important, most used one is the corn, and in our region here, you have the milo as well. So about 90% of the energy that is the corn. It's very cool because the price is cheaper. As a protein here, we don't have the most important one is soybean meal is the most important one. We don't have it here, but it's expensive. So in our business, in our research as animal nutritionists, we try to replace corn with other alternatives and try to replace the soybean meal with other alternatives as well that are cheaper and try to not reduce the average daily gain or the performance of the animal. So protein here we have cottonseed meal that is very important for us in our region, about 44% protein, and half of the protein we have in a whole cottonseed. However, we have more energy because we have fat inside here. That is very popular nowadays in the United States due to our ethanol industry. It dried distillers grain with soluble, big name that reduced to DDGS. And well, United States is the #1, the largest ethanol producer in the world based on corn. We can have this residue from this byproduct, co-product from breweries as well, but most of them in the United States are from the ethanol plants. And it's interesting because we have, well, a lot of variability here depending on the process, but range from 24 to 32, 34% of protein. But interestingly, we have a lot of energy as well. So distillers grain, in our research here along the years, we proved that we can replace for finished lambs in feedlots, we can replace 100% of the protein, for example, the cottonseed meal or soybean meal. And energy, sometimes 70, 60, 80% of the milo or corn in the diet. So it's very interesting. If you are in the region that you can be available, you can take advantage of that. And now we have here one segment that is very important as well, the minerals and vitamins. So we have a premix ready here. So it's a formulation, depending on your region, you have different deficiencies that we have to address to make a formula and have your mineral ready to the animals. And come back a little bit about protein that I was almost forgetting about non-protein nitrogen. The ruminant can utilize, for example, urea is the most common one. The ruminant can utilize non-protein nitrogen sources, and the microorganisms inside the rumen gonna produce the protein that the animal needs. So this is ammonium chloride, but it's just to mention that we have this source as well that we can use to balance the ratio. Limestone as well, and use this mix of vitamins and minerals. In some additives, in another class, additives actually is to improve the feed efficiency. We have many products or improve, for example, the shelf life of the feed, but most of the times we use to increase the feed efficiency. Well, now we have a challenge. How to mix the ingredients to attend the nutrient requirements of the animal, how to balance. So the key word here is balance. It's the same that our life. So how to balance this stuff. So let's move to another table there. So here we have mixed feed in different forms, how to present the feed to the animal. The two first here is very basic ration of his hay, based on hay, milo, distillers grains, and minerals and molasses. And this is using the same concentrate, but we change to cottonseed hulls. So this is a complete ration we call totally mixed ration, or TMR. So we serve here normally in feedlots, because we mix together the roughage from one side and different ingredients, protein, energy from another side, of course minerals as well. So to balance, depend on your, if a sheep or goat or your category that we will talk a little bit later. In this, like a supplementation. If you have your roughage on pasture, how to serve the extra that the animal needs for example, gestation, the final part of the gestation, that the animal increase the demand in nutrients, or for example, to finish animals, or flushing in reproduction. So we serve without the roughage. The roughage that you get, the graze, the browse in your pasture. But the supplement, we concentrate in this loose, like a loose feed. In this, we can have the feed also in pellets or cubes. I'm showing here some cubes. It's very practical. We have different in the market. For example, 24% protein, 32% protein. So we have to be aware at this point your availability of forage and also the quality of forage that the animals get available to eat, and to choose the right cubes to use. But, well, it's more expensive than mixed feed. Is one thing that you have to keep in mind. So, but I'm a small rancher. How to mix feed in my property? You can, for example, join forces with 2 or 3 neighbors and buy feed cheaper and mix it feed and have divided with you. So, but cubes are very practical, but have price on that. And also, we can show over there some tubs, blocks, mainly for mineral supplementation. They leak, and is interesting as well to serve in different forms. This is just to exemplify different forms, like blocks, mineral blocks, or tubes. All right, come back in here. Now we have the challenge, how to feed our animal, how to know better our animal. So the animal is a ruminant. So inside the animal we have a rumen, a complex, so it's a 4-compartment stomach. So the main important is the reticulum rumen together. In the rumen, I'm not killing, is this size or more. So we can have like 5 gallons a rumen in a sheep or more. So in the rumen, the animal gonna eat. Basically, they don't do any mastication before, and after that, long time ruminating. They're gonna remasticate and produce a lot of saliva to have the pH balance inside the rumen. And inside the rumen, you have microorganisms here. So you have to actually, to feed the animal, you have to be efficient in feed your microorganisms inside the rumen. So in the rumen, I have this scheme over here. You have, like, they mix inside, you have some muscles to mix the content inside, but you have a little bit of compartmentation here, like fine particles going to the bottom, and like the feed from yesterday going to the bottom, because needs to be, the particle size needs to be very, very small to pass through the digestive tract of the animal. Some liquid in the middle here, and the coarse particles on the top, or the feed from today. The hay that you serve today will be on the top, that will be ruminating, and the animal always has to have something inside the rumen to work well for the bacteria survive. And you cannot change the diet suddenly. One day you serve one diet, another day you tend to concentrate 100%. You can kill the animal doing that. You have to change, to switch day by day, like during at least 2 weeks, to adapt the bacteria to a new feed. Mainly when you talk about roughage concentrate ratio. And on the top here we have the gas. When the animal are fermenting, when the microorganisms fermented the bacteria is attacking the cellulose, you know,
getting the protein will produce energy for the animal, acid, volatile acid, fatty acids, volatile fatty acids here. That will be energy for the animal, and also they're gonna produce more microorganisms. They're gonna reproduce, and these microorganisms will be important for the protein. It's very good quality protein for the animal. And in this process, they generate carbon dioxide and methane. So it's a lot of gas, and they're gonna eructate these gases. Okay, and coming back now, a little bit about our the category of the animal, the category. So we have some animals over there we can show. So we know now about feeds, we know now about how the rumen works, but we have also to know what animal I'm gonna feed to calculate, to balance my diet. For example, these are our animals from our trial here. They are females. They are in the feedlot for finishing, in a finished diet, but could be replacement use. So you're gonna see, for example, if a mature ewe or a dam, how is the stage, the pregnancy, lactation, everything will be important to have the right requirements, and you calculate your ration. First, you divide your first requirement to be the maintenance. The maintenance is the nutrients that the animal needs to survive. Sometimes 70% of the nutrients that you are providing for maintenance. And on the top of that, you have to sum the amounts to pregnancy or lactation or flushing or finishing, or if you're rams. So you have to know your species, if it's goats or sheep, and your category in your herd. Let's come back a little bit here. So now in our lab here, we have this facility that is very interesting behind me. So we can put here more than 100 animals, individual pens to study the intake, average daily gain, and gain feed ratio. That is very important for us. And here in our studies, we are developing, we call physical characteristics of the forage. So I put here that to show, for example, alfalfa hay with a ground, but you can see only on the top. But this is a mixer ratio over here, TMR, total mixer ratio. You can see on the bottom, for example, the concentrate, fine particles here, on the top hay. So we are developing a new system to evaluate these physical characteristics, not only the chemical composition that I recommend you to do the test, the forage test, your feed test to see the composition. For example, but it's not only the fiber is important, the content of fiber, however, the particle size. So last year, our study was about the particle size, because the animal needs not too short to chop, too small to grind the stuff, because they cannot gonna ruminate. If they're not gonna ruminate, they're gonna not produce saliva, the pH of rumen goes down, and you can have the acidosis. That's a very important problem in feedlots. So the animals have to be something to masticate, to produce saliva, to ruminate. And we can show here, this is a Penn State particle size separator. I put a TMR with hay here, 20% hay, and the rest concentrate. You can see the different screens. We can separate and see what is the effective fiber. This is very important as well for research. And to wrap up here, talk a little bit about our current research is about corn cob. Lubbock, one researcher, Dr. Chute in Lubbock, developed a new corn and is going to release soon. Drought resistant and disease tolerant and high yield. And now we are testing the residue of the corn production, that is the cob, just the cob without the grain, and grinding and mix with other ingredients and try to see if this corn is better than the regular corn cob, and if it's better than normal ration based on hay and concentrate. And if you have time, you can come inside a little bit just
Yeah.
for a walk.
We've got a couple more minutes, and we got to go back. If y'all have some questions in the chat box, send them in. We can try to entertain some of those. But
Yeah, we can have here like individual pans. So we put the animal, we measure the feed every day, and we collect also the weigh bags. On the other day, 24 hours later, we remove this pan with the feed, with the corn cob, and we collect it and see how much the animal is eating every day. And we compare with the gain. So weekly, we weigh them and see the average daily gain and compare with the intake, dry matter intake. And we can have feed gain ratio. That is very important. The feed efficiency, that is very important. And you're gonna take more samples. You're gonna collect rumen fluid using a stomach tube, and you're gonna take the animal and harvest and see the carcass and see the quality of meat as well, because this new corn can improve the quality of meat. And just to finish here, to talk about we are the pioneers here. We founded together with other colleagues the American Consortium for Plant Secondary Compounds in Livestock Systems. We call plant secondary compounds, but actually these compounds, such as tannins, such as essential oils, they are very important for the plants. So we are now trying to study how the effects of these compounds in nutrition, in animal health, and the quality of the products.
All right. We've got a question here from Dr. Hale.
So as far as processing of corn, you mentioned that. So as far as whole corn with sheep and goats versus cracked corn versus other processed corn.
Yeah, this is a very interesting question. Well, compared with the cattle, the sheep industry is far away still. So about processing corn, when you are using like very low roughage in the ratio, like 20%, we use just cracked corn and coarse grind corn. We don't use fine grind or roller corn or flaked or nothing. But we know that the livestock, like in the beef industry, they are using flake and a lot of process to improve the digestibility of this corn in the rumen and also in the digestive tract.
All right. Well, fantastic. Thank you, Dan. That was a lot of great information to digest in a pretty short amount of time. We're gonna take a break now. We're a little bit ahead of schedule, but we're gonna take a break, and we're gonna be coming back at 9:45 at the wool testing lab. So thanks everybody for joining us. Have a short break here. Go stretch your legs. Get up and about in a bit. We'll be back at 9:45. Thanks.
Welcome back from the break. We're now at the Bill Sims Wool and Mohair, Dr. Ronald Pope and Dr. John Walker to discuss producer fiber testing services that happen here at the center. Baron?
Thank you, Daryl. What I'm going to talk to you a little bit about this morning is what happens when we receive samples from producers throughout the United States or the world. The first thing we do is when we get samples in to determine what they are, we usually primarily get side samples nowadays, but we also get fleeces. The first thing we do though with the samples when we get them in is we log them in, who they came from, where they came from, what type of fiber it is, and make sure the samples are prepaid. If it's side samples, we go ahead and take them into this next lab where we'll go in just a minute. But if it's fleeces, primarily what we do first is weigh the fleeces, because producers always want a fleece weight on their sample. After that, if they want staple length, we will spread the fleece out on the table and take 10 random staples from that fleece, sack them up and for subsequent analysis in the next lab where we'll go in just a second. After that, we will take the fleece and put it in this instrument here, which is a core machine. And what we do is we take 2 25-gram samples from that fleece to determine yield analysis. And after we get these 2 samples, we will then transfer them into the bigger lab where most of our work is done. And that's where we'll go right now. Okay. About 4 different inches of fiber diameter. And I said a while ago that we primarily get side samples for analysis. One of these machines we have is called an OFDA. It's a portable OFDA 2000. And this is primarily what we analyze side samples on for fiber diameter. We have another instrument over here, which is an OFDA benchtop. It's not portable. And what we analyze here is mini core samples that come from the yield analysis of the test we perform. Both of these 2 machines do the same thing. Only difference is this machine right here will give you a staple length on the side samples, whereas this one doesn't. And we can interchange samples. We can run side samples on this machine and core samples on this machine, but we try to keep them both the same. I mentioned that we obtain staples out there if they want staple length measurement. These staple lengths, I have some put out here on this table. These are the kind of staple lengths that we get. What we do is scatter them out and just take them on here on this little pad, stretch them out and measure these staples. And it's 10 of them we obtain from a fleece, and we measure them to the nearest 1/4 of an inch. After we get this 10 staple measured, they are then taken into another room. The measurements are put on a calculator or a computer, and we determine the average staple length, the standard deviation, the coefficient variation. So as mentioned previously on the yield samples, now have the 2 25-gram samples, and what we do here is we wash these samples. We mix up a solution of soap and sodium carbonate. We wash the samples, which takes about 10 minutes to do. After the samples are washed, we transfer them over to these ovens where they're dried at 105 degrees centigrade overnight. We take them out, reweigh them, and from there we obtain what we call a bone dry yield, and convert that to a lab scarred yield by adding in moisture content. Now, if producers want a clean fiber present, there's 3 other tests that we do, and they're performed in this lab. The first test is the grease and we have the sample put into a thimble, a cellulose thimble. We use alcohol as an extraction. After that, we boil down to the residual grease, and all we have to do is weigh it back to get a percentage of the residual grease content. The next
Y'all stand by. We've got a little technical difficulties there in the wool lab. We're going to reconnect and try to rejoin in progress.
Ready?
We're back on.
Okay. The next test is the percentage of ash content. What we do is weigh out about 5 to 7 grams, put it in one of these little crucibles. We char it down under the hood. After that, it is transferred over to this ashing oven. This crucible was placed in here for 6 to 8 hours at 700 degrees centigrade. And after that, we weigh back the sample, which is just dirt particles that are in the wool. And therefore, we can calculate the percentage of ash. The last test that we run is vegetable matter. And what we do here is mix up a 30% solution of soda, a caustic soda solution, heat it up to boiling. Then we take a sample, an old sample here, weigh it out, we pour it into this solution and let it dissolve. What it will do is dissolve the fiber that's in that sample, and the remainder will be the vegetable matter content. After that's through boiling for 3 minutes, we take it off, neutralize it, wash it several times in the sink, and then add a little sodium hypochlorite or bleach and dissolve the remaining if there's any left. And from there, it's just a matter of calculating the percentage of vegetable matter content left. Now, that pretty much is all of what I'm going to talk about as far as the analysis. We Shetland wool samples in the last several years. Our wool, our white wool or merino type wool or Ramblais, we haven't done a lot of those samples in the last year because the numbers are well down, but we'd like to get back into that. We also do a lot of producer grower samples throughout the year. We have the capability of analyzing wool samples for NSIP. That project goes on there. Every year we still have a goat test that we analyze the samples for, the fiber samples, and as well as the fecal samples for the quantity of juniper they're eating. Every year universities send down their judging teams with their fleeces, and we help them with determining the yield and fiber diameter of their fleeces so they can practice on them for the upcoming year. The last thing we probably do during the year is during stock shows, we assist with different contests. Primarily the one here in San Angelo is we have an Angora goat futurity and a yearling ewe futurity, and we do all the fiber analysis for those two contests. If that concludes what I have, unless there's some questions or something. Otherwise, I will turn it over to Ronald Pope inside.
Well, good morning. Good to see all of your smiling faces out there. We can't see them really, but anyway, my name is Ronald Pope. I'm going to discuss a little bit about testing wool for commerce. Farron just did a great job of explaining all the abilities of this lab to provide information on wool quality and quantity for growers based on individual animals. And the importance of that is this lab can do those things right now, especially for growers that are involved in the National Sheep Improvement Program, wanting to look at different wool traits in their selection program. And so anyway, we can do this for wool and mohair, alpaca, cashmere, almost any fiber that animal produces. But again, I just want to encourage growers to consider this lab here when it comes to testing individual animals to be used in a genetic program. What I'm going to visit with you about is testing wool for commerce, which is a totally different objective than individual animal assessment. When we talk about testing wool for commerce, we generally use the term, well, I got a core test result on my wool this year. That means that the wool that they harvested and brought into a warehouse or marketing it and sent it off to a test lab to get a report or a certificate that a financial transaction could be made regarding those results. And so the two main things when you look at a core test, the two things that we are able to do are average fiber diameter and an estimate of yield. And Farron just got through going through some of the instruments we use here regarding how we determine average fiber diameter and yield. Fiber diameter obviously is important to the processor when it comes to what that wool's intended use can be. In other words, what type of product is it going to end up in? And so it's very important that they fall within a certain specific fiber diameter or micron range for their intended use. The yield tells us basically how much wool is in that greasy lot of wool. And so we do test wool for commerce to allow us to determine what is a fair or be able to negotiate what is a fair price that's beneficial to the grower, obviously, but also to the one that's buying the wool. It removes a lot of the unknowns. And so it allows a sound decision to be made of that transaction. What has happened in the past, we had a test lab, one private test lab here in the United States that operated for over 60 years, and they're no longer in business. They ceased operation in the start of 2020. And at that time, we had the wool industry here in the United States request that the Bill Sims Wool and Mohair Lab implement a commercial testing lab for core test. Over that 60 years, there were a lot of advancements made in technology and to enhance, speed up the testing process without compromising the integrity of accuracy and precision of what we're measuring. And so one of the instruments that we use in commercial testing or testing wool for commerce is the laser scan. The instruments that Farron showed you earlier are they're precise and accurate, and they can do a good job, but they're not certified to be able to do testing for commerce. We'll step right in here. We can look at the laser scan, and I'll just leave this screen up. I won't run through a process on it, but if you'll come on in, if you can get a shot of that screen. Basically, this instrument here will give us a fiber diameter, standard deviation, curvature, various other locked up. Back on that. Are we up? Okay. Sorry to lose you there. But anyway, the laser scan is a new development. It was developed in the 90s. Prior to that, we used a projection microscope that we could count or measure about 100 fibers every 15 minutes. This one will measure over 1,000 fibers in a matter of 30 to 40 seconds. So obviously, we're getting a better precision when we're using this machine. And so the other component that we look at when it comes to testing is yield. And not much has changed in that for the preparatory areas of scouring and drying, obtaining some of this newer equipment that will speed up the process. And I've got a little video loop there that if they'll engage it.
Samples are washed in laboratory-sized scour bowls to remove dirt and grease. The scouring process involves a number of cycles and is standardized for water volume and temperature, as well as the type and volume of detergent used. The samples are then dried until no moisture is present in the wool. Once the samples have been washed and dried, they are weighed to determine the amount of clean wool following the washing and drying process.
Video over. First part of that video, we're going to get 2 3-bowl scours that will basically reduce the number of people and the time. If we were doing the same amount of scouring, it takes 6 people. This operation, new equipment, will allow one person to do it, and it'll be automatic and be faster than just hand washing. The drying is also the same. It speeds it up rather than having to dry for 8 to 24 hours. It'll dry those samples in 4 hours. The goal is to be commercial. In other words, being able to get core tests in here, get them run, and get them out within a day or a day and a half. And so just to give you an update on where we are with the commercial lab, we are still in the process of trying to procure these pieces of equipment. It'll take 6 months after we order them for them to be manufactured and then shipped over here. Getting them installed and in place and getting people trained on the proper operation of them will take more time. So we're looking at the late 2021 before this lab will be up and operational to test wool for commerce. If you have questions, I know we're out of time, please do not hesitate to contact any one of us here at the Research Center and let us know what your questions are. Thank you and have a good day. Thank you for participating.
Okay, I'm John Walker, and I'm going to show you our near-infrared spectrometer and how we use that to test wool. So this instrument is a near-infrared spectrometer. What it does is it shines light on samples, and it measures the amount of absorption in various wavelengths from 500 nanometers up to 2,500 nanometers. The longer wavelengths are what we call the infrared. So I want to show you some wool samples here, and can you give a close-up on these? You saw all the stuff they were doing in the other lab. You can look at these samples and you can guess at what you think the average fiber diameter is and the yield on them. And I think it should be somewhat obvious which one is going to yield more wool, which one is less dirty. So for this, we're going to run these two samples, and I'm going to show you how this works. All you have to do is take a staple and load it into this slide. And it's ready to go in the machine, okay? So I've already made this one up. I'm going to put this one in. Again, this is this sample. All I have to do is tell it to scan. Okay, 30 seconds. First thing the instrument does is it has a ceramic that it collects reflectance off of to kind of standardize it. And in a minute, that slide's going to drop down in there, and it's going to take 36 different scans on it and collect the different absorbance of that material. Now, the way near-infrared spectroscopy works is that molecules have chemical bonds, and those chemical bonds vibrate. And if the vibrational frequency of the chemical bond is the same as the vibrational frequency of a particular wavelength of light, it absorbs that wavelength of light. And so you can see here, this is the spectrum we got off of that. And where it's low, it absorbs more. And as it goes up, peaks and valleys, it absorbs less. But in that length of time, what we got was the yield on that sample was 46%. And the average fiber diameter was 19.5 microns, okay? Now I'm going to stick the next sample in. So the calibrations on these were developed by taking side samples, and that's what these samples right here are. They're just side samples, staple side samples. We collected spectra off of them. We cored the fleeces that those samples came off of, and we ran the standard lab test on them. The advantage of that is normally we just do side samples. And we hope that that side sample represents the entire fleece. Our calibration methods allow us to actually take the chemical composition of this fleece, of this wool sample, and calibrate it back to the whole fleece. So it's actually a better representation of what that fleece is than just a side sample. And I'll show you some of that in a minute. Again, you can see here's the spectra. With your eyes, you can't see a whole lot of difference. But if we look at the predictions, the yield on that fleece was 62%. Remember, the last one was 47%, and the average fiber diameter is 20.5 microns. So slightly coarser, but much higher yield, meaning that that fleece is really more valuable. We are also going to use this instrument when we get the commercial lab going. The soxolate procedure that Farron talked about, which takes 4 hours to run a sample, we can do it in just like we did this, and we can get that residual grease. The ashing, that also takes several hours, we can, at the same time, just like we get the yield of average fiber diameter, we can get residual grease and ash just by running that sample through this instrument. So let's see if we could bring up the slides, or if you want to, do you want to just do this one?
It's probably going to be better for the slides.
Okay.
And the slides on there.
Slides are okay. This first slide, what this shows is we took a fleece core sample, and this is the yellow bar to the left.
is a core sample from a fleece. We ran it through the lab. This is to get the lab scourge yield. We took a side sample, just like the samples you were just looking at. That's the green bar. We ran it through the lab. And then we took that same side sample, a subsample out of it, and we put it in the NIR, and we calibrated it against that yellow bar. So it's not surprising that when we look at lab scourge yield, the side sample on the NIR is, on average, gave the same yield as the lab scourge yield. The yield on the side sample was actually lower than the yield on the core sample. Okay, but here's what's even more interesting. If we can have that next slide, this just shows the average amount of error on the two methods. So if you send a sample to New Zealand, a side sample to New Zealand to get yield, we're going to wash that side sample, and just like we've been talking about. And the point being that it's not going to be as accurate, and it's going to have more error. So the average error on comparing the lab run or the NIR was about 1.6% on the lab, and for the side sample it was about 8.8%, so half as much error. If you go to the next slide, again we're looking at average fiber diameter, and we see the same thing basically. If you look at just the average fiber diameter, again the standard is this core lab on the left, the yellow bar. The NIR gives you the same value. The side sample is actually coarser by about a micron. If we look at the error, that last slide in there, again, there's less error in the NIR than there is in the standard lab procedure. So this very powerful instrument, and what the nice thing about it, it's going to allow us to test grower fleeces for individual animals to make genetic selection to improve both their yield and the average fiber diameter on their fleeces. And that's all I have.
So we're scheduled to take a break.
Ready?
Yes.
All right, welcome back. Reed Rudden here again with Mr. Bill Costanzo. We are at one of our livestock guardian dog bonding pens. So we're out in a pasture here at the research center. And a few years ago, we built these livestock guardian dog bonding pens to do some research. And so one of the things that we wanted to investigate is if we start bonding these dogs out in the pasture versus up near the headquarters somewhere, what kind of improvements can we see in those dogs bonding to the animals and going forward? So predation, as many of you are very aware, is one of the biggest problems in the sheep and goat industry. And, you know, we've known for thousands of years that livestock guardian dogs can be a very effective tool to prevent predation. But unfortunately, there hasn't been a lot of conclusive research done about livestock guardian dogs, and especially here in Texas. And so we've been doing some guard dog research for a number of years now, and this is one of the areas where we do that. So our bonding pens, if Corey can kind of pan out here, we're out in roughly 100-acre pasture, and within this pasture, we're just offset from the fence a little bit, but we've got a 1-acre bonding pen. And then within that bonding pen, there's a 60-foot by 60-foot exclosure. So when we start with a group of puppies like these two here that are kind of playing in the shade, they come in and they start in this 60 by 60 enclosure so that we can create contact. There's some sheep in the back, but they're a little bit scared of us, so they're on the back side over there. Hopefully you can see them. But these dogs have constant exposure to them, and so we'll do this for a couple of weeks till we start seeing that the dogs are bonding well, and Bill will go through kind of our bonding strategies here in a minute. But we have 4 of these different bonding pens in 2 different pastures. So there's one right here, and there's another one about a quarter of a mile away that's within the same pasture. And so they'll stay in here until they're about 2 months old, and then we let them out. But it's not just a concept that we're looking at. We have a couple of treatments. There's different thoughts that maybe these dogs should be raised by themselves with animals during the bonding phase, or there's thoughts that they should be raised in groups where they have other dogs around because they're going to have to work with other dogs in the future. And so that's our projections going forward. We're raising them as a single or as a twin. And then we also have electricity in some of these. So we have an electric fence, solar charger, and there is an electric line that's run around the inside of these pens right at the bottom. So when these pups are young and they're growing up, they'll get bit by that electricity. So we're trying to teach them to respect fences. And so if you've ever had guard dogs before, or if you've ever been around them, you'll know that one of the challenges is keeping them in the pastures where they're supposed to be and getting them to bond with the animals and stay where they're supposed to be. And that's what some of our research is really focusing on, because they can be very effective when they're where they're at and where they're supposed to be. So this is a team effort. I'm involved in the livestock guard dog research team. Dr. John Aguacheck has done some research in the past at one of our ranches in Menard. But again, we don't just practice it or preach it, we practice it. We've got dogs here. We've got dogs at the Menard Ranch, the one in Sonora and Ozona. And so big ranches where there's not always somebody with them all the time. And Bill leads our livestock guard dog efforts and manages the dogs and manages the day-to-day operations here in these bonding projects. So with that, Bill, you want to kind of tell them what the process is that you go through when we're bonding a set of pups, and then, you know, measuring their effectiveness thereafter in their lifetime?
Sure. So we start out, we don't breed dogs at all. We buy our pups from breeders here in Texas. And so we originally started with 6 pups in the first go around last summer. They were all 8 weeks old. And so as Reed said earlier, we started with 2 pups in one pen and a single in another. And one pasture had no hot wire, and then the other pasture had hot wire in it with those pups. And so we tracked those pups over time. And so they spend about a month in this 60 by 60 enclosure. And then we open this up once they've bonded well to the sheep and goats that we have. And then they go into the 1-acre pasture here until they're about 5 months old. And then at that point, we release them out into the 100-acre pastures together. We put GPS tracking collars on the dogs and all the livestock. And we track those dogs for several months. And so what we found was that the dogs that were bonded in pens that had hot wire were less likely to leave the pastures than the dogs that didn't get bonded with hot wire. And that is held true now, even we're about 7 months out with those dogs being released into our research ranch and a cooperating producers ranch. And that is holding true still when those dogs are almost a little bit over a year old at this point. The dogs that were bonded with hot wire are not leaving the producers' pastures, while the dogs that are on our research ranch routinely leave the pasture, but then they do come back at some point in time.
But Bill, you want to explain a little bit about the individual personalities that these dogs do? And like, we're doing research where we've only had a limited number, and that's one of the things that I think producers have a limited number of dogs that they may have. They may have only had experience with, 2, 3, 4, 5 dogs in their lifetime, and each dog is very individual. And so while this is showing some promise now, our goals is to get dozens of dogs through this program and measure them long term. But can you tell us about the individual personalities that you've seen in the dogs that you've raised and bonded so far?
Oh, definitely. The first set of dogs that we got were Maremma pups. Those dogs were a little bit more shy in general than if you follow our Facebook page, the superhero pups that we had. And so the Maremmas are the pups that have not left the pasture at all. And they were a little bit more shy at the beginning. I do socialize the dogs. I think that's very important, and I highly recommend that to producers. So basically what I do with the puppies is, at least 4 days a week, I come out, I visit each one of the dogs, I handle the dogs. We also leash train the dogs. They get tethered for a certain amount of time, and then it increases each week. And so the idea of that stuff is that you can easily be able to catch the dogs later on in life. It's important for doing health checks and just maintaining the dogs, or be able to easily move the dogs to a new pasture. So socialization is a big thing that we promote here with the dogs. And as Reed said, each one of the dogs does have their own personalities. They're just like humans. Even between these two dogs, Bo, the one with the black collar, is a lot more friendly than Luke, the one with the orange collar. And so they are, yes, as you can tell, Bo's coming over to visit with Reed. They definitely do have individual personalities. And some of the dogs, we noticed that they prefer to be with goats in the first round, and the other set of dogs prefer to be with the sheep. And so it really just kind of depends on the group of dogs, what's best for them, and to be able to fit that to your operation.
Yeah, and the reason we really got this, I don't know if I stressed it early enough before, but these livestock guardian dogs, they have the genetics to guard and do their job, but the way that they're raised in that first 16 weeks or so of life really influences what they do. And if they're raised in the absence of humans, they can become feral and very difficult to manage. But if they're raised near the house with people all around all the time, even if they're with livestock, they may become too assimilated to people and they don't want to stay with their livestock. So it's that happy medium and that happy balance that we're striving for, and that's what our program is trying to demonstrate and prove through science and research. So we got any questions coming in the chat box? Checking them on them here. Any questions from the gallery? Any more thoughts there, Bill?
Just one quick thing on the socialization. People ask me all the time, like, well, how much time do I spend with the dogs? As far as like socialization of brushing the dogs, checking their ears, their teeth, their feet, I spend about 5 minutes a day, usually about 4 times a week. So it's a very small percentage of time. And I can tell you that we can still catch all the dogs that have been released so far through the bonding program with just that short amount of time socializing them. So it doesn't take a whole lot to be able to catch your dogs out in the field on a regular basis.
Yeah, good. All right, well, we're just about out of time, but we're going to transfer and transition over to our next session. So, we try to focus on the highest issues or the top issues with predation and with guard dogs. One of those is that bonding and keeping them where they're supposed to. So we're working on that. But another one is feeding the dogs. How does the best way to feed the dogs so that, you keep them healthy, keep them gentle, but then they also, we're not feeding all the livestock and feral pigs and raccoons and everything else. And so we've got a whole lineup here that Bill's going to go through and kind of pros and cons of each of the feeding scenarios we've trialed out. So Bill, I'll step out and let you take it from here.
Thank you, Reed. So basically there's two different types of feeding systems. There's the basic hand feeding system, and that's just as simple as a bowl. And you feed the dog on a regular basis, hopefully every day or every other day. But with each of these methods that I'll talk about, there's pros and cons. And so the nice thing about hand feeding your dogs individually is you get to see those dogs every day. You get to put your hands on those dogs every day. But the problem with that is if you're a large producer, is finding the time and just being able to find the dogs on a large ranch. And so for a lot of large producers, hand feeding is just not an option. So the other option is going to a self-feeder. And so I have a variety of self-feeders here for you guys today. This is just a real inexpensive self-feeder right here. We've made some improvements. We've added handles, a latch, some hooks on the back of it. The next one here is a timed feeder. This one runs off of solar. If you're looking at a timed feeding feeder for your dog, I'd highly recommend the solar one. It just makes life a lot easier for you and you don't have to change out the batteries. But there's pros and cons to even these two types of feeders. The self feeder, your dog can get feed whenever it wants, okay? But also every animal, that's out there can get dog food whenever it wants to. And so your feed bill doesn't really decrease with this type of method. If you use the timed feeder, okay, you can set the timer to go for however many times during a day that you want. And it can go from anywhere from usually just 1 second to about 30 seconds to run. The issue that you have with the time feeder though, is if you have multiple dogs, it's making sure that all the dogs are actually there to get the feed. And if they're not, then the feed just sits there. And again, you have a feed source for varmints to get food. And so, one of the things that we've been researching is try to, keep non-target animals from getting the dog food. And so the two things that we've looked at are just taking the simple inexpensive feeder and adding an RFID doggy door to it. So basically how it works is the dog would wear an RFID tag on its collar. As the dog comes up, the door releases, and then the dog is able to eat the food. The concept was really great with this, but the problem is that on an inexpensive RFID system, the metal in the feeder interferes with the RFID signal. And so what was happening is it was fine for the puppies because the distance was close enough for them to be able to get their head in and release the door. But on an adult dog, which is about 12 inches or so away from the door, it wasn't able to read the signal because of the metal interference. So our next plan that we came up with, and this is what we're just trying out currently, is adding a doggy door to the gate of our feeding stations. And so it's the same type of system. This one's obviously much bigger for adult dogs. And so the dog, again, wears a tag. When the dog comes up to the door, it releases, they go through and go into the feeding station. And so this is the one that we really hope will end up working out. It will be able to keep out, any animals other than your dog from getting into your feeding station, and it should really reduce your feed costs if we can get this system worked out for you. So please check our Facebook page for updates on the RFID doggy door system. So we have two different types of feeding stations here today. This is a basic, just kind of simple, inexpensive feeding station. And so there's a feeder inside. This is just a basic wooden jump gate. You can adjust the height of this to meet your dog, but I like to see the dogs jump up and go through. To me, having them crawl underneath, which is definitely an option, but I just kind of think that promotes the dogs crawling and digging underneath your fences, which we don't want them to do or to go wander. And so the dog just jumps up and goes through here, eats, and then leaves. The problem with a feeding station like this that we've had, we do use this station here. If you have a lot of feral hogs, the hogs tear up these stations really easily. And then they get into the food and they tear up your feeders, and it's just a constant issue. And so what we did is I looked at a bunch of different what producers were doing. And so I came up with this design that we have here. And I just kind of made it a little bit beefier than what was out there. And so this is definitely doesn't get destroyed by the hogs. It's a pretty heavy unit. It does take a couple people to move. We've put these out at our research ranch in Menard, and we've definitely seen a decrease in our feed costs. The paneling here is adjustable. And so for the puppies here in the bonding pen, I start out with the bottom of the gate open. And then as they grow, I gradually move these panels down and I open it until it's up to this third spot where they can just easily jump through. The nice thing about this unit is it's easy for you to get inside. The feed's pretty well protected. It does provide some extra shade for the dogs. A lot of times the pups in the bonding program will either be inside the feeding station or outside of it in the shade. This is also really great for a bonding pen because it gives you an area for the dogs to get away from the livestock. So if they're being bullied at all, the pups will go inside here and they'll kind of hang out and until they're ready to get back out and go socialize with the livestock. So these are two of our feeding stations. There's I can send plans if you guys are interested in that type of thing. Just message me through our Facebook page. There's all kinds of different ideas out there. Producers send me different types of feeding stations. You can put these on trailers. Just whatever you can kind of think of will probably work. The main thing is that you want it to be definitely secure and to be able to keep anything else out of it other than your dog. And so what we've done is that the RFID doggy door feeder over there, we've adapted that, and it just mounts right on this same type of feeding station that we have here. And we'll be giving you guys some updates as time goes on with that project.
So Bill, got a couple questions coming in. The first one is, where do you place the hot wire? And what have you learned so far with where you place the hot wire and how it keeps the dogs trying to get out or not?
Okay, so the hot wire in all of our bonding pens is, I have an extender off the main fence, and it's between 4 and about 6 inches, usually kind of depending on the terrain of the ground, is the height that I have it up off the ground. And that works pretty well. I have it at that height so that when the puppies are small, they can't get their heads underneath it and dig out. So it makes sure that they get shocked if they come up to the fence. And I do move it up a little bit as the dogs get older. If I notice that they're digging or anything like that, I will adjust the hot wire as the project goes along.
Good. Another question is, what is the minimum amount of goats needed to bond to puppies? And you might expand on that. Is, do we use the same animals the whole time?
So what we've done is we have generally in this 60 by 60 and then into the 1 acre enclosure, we start out with between 4 and 6 head. And so what we did with the first round of pups, and we'll do with the second round that we have coming up here in September, is we have a mix of animals. So we have Rambouillets, Dorpers, meat goats, Angoras, because we don't know where the dogs are going to end up going once they leave the program. And so we want them bonded to all the different species. And so the other thing that's important as you go through is every 2 to 3 weeks, you really should switch out the animals. Okay, so if you have a mixed group of animals like that, you move to a new mixed group of animals. Or if you're bonding all the Dorpers or all the Rambouillets, you still switch them out, because what you don't want is the dogs to bond to specific animals. You want them to bond to a species of animals. Because if the dog bonds to a specific group of animals, and for whatever reason, then you sell those animals, the dogs don't know what happened to their pack, basically. And so then they start to look for those, and then they start to wander. And, that's obviously a big issue with the dogs, is wandering. So try to bond them to a species as a whole instead of specific animals.
Or like the case we've had in Menard, where they were bonded with a group of sheep, and then you put another group of sheep in there, and they don't provide protection to the second group.
Yep.
And so then you'll still have predators come in because they just don't have a dog with them all the time.
Yeah, so it's important when you take the dogs to a new location, or if you're adding more animals, to do a short bonding period of those animals together with your dogs.
All right, we're going to transition over a little bit. Hopefully we don't get too much of the home video action here. But one thing that I failed to mention earlier, and it's very important, is that Bill Costanzo and some of this research is all funded by the Texas Predator Management Board. So most all sheep and goats sold in the state of Texas, there's a checkoff that goes to the Predator Management Board. And they've seen some of the research that we've been doing, and we've had a cooperative agreement with them, and they're funding some of this work because they see the value in it. So, it's important to do collaborative work with the industry, and that brings up our next panel here. And so one thing that we've, had some concern with is there isn't really a representative group that represent livestock guardian dogs, whether it's for breeders or for the producers that have done it. And so we've reached out to the industry, and very fortunately a group of people have stepped up and are in the process of developing the Texas Livestock Guardian Dog Association
So the industry has representation for the use of these dogs, and it's also a means for breeders to advertise and things of that such that's being developed. And we have these two gentlemen here. We have Walter Pfluger and Michael. How do you pronounce your last name? I always get it wrong.
Annunziato.
Annunziato are some of the founding board members that's getting it going. And so they're going to talk to you a little bit about why they see value in this and why they're helping getting this going. So thank you, gentlemen, for being here.
Good. Thank you, Ray. Well, I'm a producer, not a breeder, and practiced law for a number of years, and last year decided to retire from the law practice and get really back into producing sheep and goats. And I thought, where do I start? And I went to the sheep and goat raisers meeting and listened to Reed and Bill talk about their program, a very exciting program. And then they introduced the concept of a breeder and producer organization that was separate from their program. We've been working all year long on that organization. We came up with the name of it with a group of people, a group of breeders primarily. And we formed a Texas corporation, the Texas Livestock Guardian Dog Association. We're in the process of working with a CPA to determine the best tax type of status that we can achieve for the goals of the organization. And I would think in the next month or two, we will have that settled, working on the different classes of membership for the association, maybe a breeder's membership and a producer membership level, maybe some different dollar amounts for those. And COVID interrupted some of the field days that we wanted to have membership drives, but I feel sure that's going to take care of itself as soon as possible, and we can actually have some face-to-face meetings, tell people what we're trying to do, and create a membership group to go forward. But I'm really excited about it as a producer, because when I first started out, I really didn't know how to determine which breeders might be in the basket of breeders that I wanted to work with. I had a lot of questions and not so many answers. And over the past year of trying to set the association up, I've met breeders. A lot of things have cleared up in my mind, and I still have a whole lot of questions that I'm excited that this association and the research that Bill and Reed and their team are doing will be helpful to me. I agreed to serve on the panel before I knew it was virtual, and usually panels get questions from the audience and don't have to say too much on their own. They just answer questions. But anyway, today we're kind of questionless, I guess. Mick Annunziato has lots of answers if you have questions.
I am a part of this organization as well. I am a breeder and a producer. One of the needs that we felt that was educating people on the dogs, what they can do, what the benefits are. People always have questions, and without a reputable place to go get good answers, it makes it hard. There's always a learning curve. We're also looking at some legislation down the road to be able to assist with for the producers and the breeders when it pertains to livestock guardian dogs.
So as a breeder, Mick, you've been doing this for 25 years, and what kind of issues and what parts of the country do issues come up more between the people that are using the guard dogs and the communities around them that might, in some cases, be real close to each other? And can you say what your experience on that has been?
Sometimes your small ranchette equations or your more populated areas, your dogs are closer to other people's animals, and people are concerned about the dogs being aggressive towards people and other animals. That is a problem, or that is an issue that we would like to be able to address and help people out with.
And probably in your experience, there's a lot of concern when people see dogs running around next to them with sheep and goats, and people who are uneducated about the temperament of these dogs can get concerned very quickly.
Temperament, yes. People don't understand these dogs don't have to have shelter. They don't have to. It's 32 degrees and 6 feet of snow on the ground. That dog's on top of the snow, basking in the sun, having a good day. And people think that dog needs to be in a heated environment. And on the same aspect, 100-degree day with the sun burning down, a water tank is just as sufficient as shade for that dog. People don't understand those things, and that's what an organization like this can answer and help people with, and lobby legislation not to cause problems with those dogs doing their job accordingly.
Yeah, I think that in my situation, I've got I'm more rural, and most of my neighbors are ranchers, and we always notify them that we're going to have dogs next to them in the pastures. But I can see that within close to the city limits somewhere, and you have people that we would like to educate more about what we're doing, the treatment of the dogs, the temperament of the dogs, and you raise Maremma dogs that you socialize, and their temperament is very gentle, very people friendly, correct?
A lot, yes. They're tend to be a more docile towards human interaction. They interact better a lot of times. Any dog that is properly introduced and has human interaction can be a dog that has good temperament, whereas a dog that is strictly left to livestock out to large open places may be a little bit more feral, a little bit more on the wild side.
Well, the mission statement for our association is to support, promote, and advance the sustainable use of effective livestock guardian dogs by farmers and ranchers. Our vision is to increase the effective use of livestock guardian dogs. And our core values, we strongly believe guardian dogs are one of the most cost-effective and sustainable means to protect livestock and crops of farmers and ranchers. We believe that sustainable use of our resources in the land that they are on is critical to the long-term welfare of the agricultural industry, and we promote honesty, integrity, and professionalism within the livestock guardian dog community in the agriculture industry. And this is what has excited me as a producer. I am not a breeder of dogs. I don't have any plans to sell dogs or anything like that, but I do have about 20 guard dogs right now in different pastures. And this is what I'm excited about. I don't think in my part of the country that I could raise kids or lambs without guard dogs. That's been my experience. And so I'm excited that we're bringing a group of producers and breeders together where people can have a clearinghouse for obtaining dogs, obtaining answers, meeting with each other and trying to solve the problems of the industry as sort of a parallel group to go along with all the great research that Reed and Bill are doing here at the AgriLife Center.
Hopefully by the time of our next field day, we'll be ready, set up to take memberships and start supporting producers and breeders with this. And there is a website or Facebook page. Bill can probably load up through this and direct people towards this as well.
I bet we're close to the end.
We are. Thank you guys very much for coming out and talking today.
I have a few questions.
We just have one question.
Yes, go ahead.
Do y'all sell the dogs after the research is completed?
So what we've been doing is we find a cooperating producer. And so part of that project that we've started is that the producer takes the dogs, they take care of all the health and the feeding and that type of thing. And at the end of the project run, if they would like to purchase the dogs, they can then purchase those dogs. The first cooperating producer that we had was so happy with them. He bought the dogs within 2 months of using the dogs, and he had never had dogs ever before. And they're very happy with them. And these two pups that are here in the pen today are actually two of their dogs that I'm doing a short little training course on for them. So are there any other questions?
Yeah. Do you have any fence signage to inform others that dogs exist in the pasture?
Yes, we do. I know that's an issue for some producers. And so all of the Texas A&M ranches have signage that say there's livestock guardian dogs on duty. That's really up to you as an individual producer on how you want to look at your liability, and how your ranch insurance is set up. Okay, well, thank you guys very much. We'll be going to a short break, and our next session will be starting, I believe, at about 11 o'clock.
To support agriculture and the hardworking people who feed the world. And for over 60 years, Ag Workers has steadily grown by holding true to principles of loyalty, fairness, and service. And today, Ag Workers has expanded by providing access to opportunities that enrich the lives of its membership.
Ag Workers is a lot like family, and you trust your family. I trust the employees, I trust the service. People love Ag Workers because of the relationships that we have with those people.
People don't thank you for your insurance. Our people do. We work for them.
They truly do look out for what's best for their customers and try to provide the absolute best service that they can.
Built on a solid foundation of strength, trust, and security, Ag Workers is more than just a business. It's people taking care of people. Ag Workers Insurance, a legacy of trust since 1948.
Good afternoon. My name is Gerald Hobson, and I am with the Bear Range and Pasture Group. Want to wish everybody the best of luck during this virtual Texas Sheep and Goat Raisers Association meeting. And we have introduced two key products into the range and pasture markets in Texas this year. Angora is a new tool, new technology, new chemistry, new molecule that's going to help those that have problems with mesquite and weed-setch manage those problems better than they ever have before. And then in the warm season perennial grass markets, we're currently introducing Resilon, which is going to help manage annual grasses and broadleaf weeds on your warm season perennial grasses. So if you need more information, contact me or Dr. Alan McGinty, who lives right there in the San Angelo area, or any of the others of our range and pasture team. We'll be glad to help you. Once again, good luck to you, and I hope you enjoy the meeting.
It's hard to let something that's been in your family for generations go, so it feels good to make a go of this. I'm Tyler Black. I'm a fourth generation farmer here in Bailey County, Texas. We grow corn, milo, wheat, raise cattle. Working with Capital Farm Credit has been great. I mean, you're not on your own when you go into Capital Farm Credit. They treat you more like family. One of the best things about Capital Farm Credit is they speak our language. Together we're much better.
The American Goat Federation represents the interest of every goat owner in the United States. AGF travels to Congress each year to present producers' concerns and takes part in USDA and other agency meetings throughout the year to make sure the goat industry has a voice in decisions that affect goat producers. In addition, the Federation provides accurate information to the general public about goats and goat products.
Eco-Deer, ecology, economy, results. Brought to you by Palmer Feed. Family owned and operated since 1943. For more information, visit our website, palmerfeed.com.
No one ever said sheep and goat ranching is easy. Day in and day out, it's a tough job. Part of our mandate at Lakeland Farm and Ranch Direct is actually listening to our customers and finding solutions that actually work for you and your farm. Whether it's handling, trimming, sorting, or crowding, Lakeland has the sheep and goat equipment to help make your day just a little bit easier. Our Deluxe Spin Trim Chute is a farm favorite for many sheep and goat farms that need an easy way to trim, tag, and vaccinate their animals. The working chute and crowding tub is a great system to help crowd your sheep and goat and make your operation an efficient one. The Lakeland Easy Clamp is the go-to product for high-volume sheep and goat handling, perfect for drenching, vaccinating, tagging, and more. Whether you need handling equipment, fencing panels, sorting attachments, or waste scans, Lakeland can help build a custom sheep and goat system that works for you and your farm. Through our farm direct delivery, our easy-to-use premium products, or our exceptional customer service, Lakeland Farm and Ranch Direct is the only provider of sheep and goat products that make life easier for today's rancher and the hardships they face.
Hello everyone, my name is Wade Polk, and I'm the branch manager at Central Texas Farm Credit in San Angelo. We are proud sponsors of the 2020 Texas Sheep and Goat Expo, and we want to help you with your financing needs. We finance and refinance farm and ranch real estate loans, as well as providing loans for operating, equipment purchases, and livestock. We understand the ins and outs of agricultural financing, and we can customize a loan to fit your specific needs. To learn more, stop by and visit with us at 1021 South Cohenheim, or visit us online at ranchmoney.com, or call us at 325-658-8545. Thank you.
It's more than a piece of land. It's a place where you create memories and leave a legacy. It sounds like a dream, but at Capital Farm Credit, we'll help you make it a reality. As the most experienced rural lender in Texas and a cooperative, we're with you every step of the way. Your goals are our goals, because no matter what your dream might be, we're all in this together, and together, we're better.
All right. We good? All right, welcome back. This is our last session of the field day before we break for lunch. To kick this session off, I'm going to talk about an app that we've had developed here at the Research and Extension Center. Bill Thompson, unfortunately, can't be here today. He's been kind of the lead person. He and I have navigated the development of this app. It was released last summer, so if you were at our last field day, you would have heard a talk on it. And also our recently retired geneticist was the one that helped us put the data together. But what this app does is it gives a relative prediction on the value chains of sheep and goats if you were to retain them and either keep them in a pasture or bring them in and feed them like what we've done behind us here. So the app is available only for iPhones. Unfortunately, we haven't had a developer develop an Android version yet. So this is what it looks like if you develop it. If you're looking in the app function, make sure you search lamb goat market forecast. If you put the word "and" in there, for some reason it doesn't show up. So you can put the "and" symbol or just leave it out and it'll pop up. Download this, and this is your front page. So on the front page, if you're going to sell lambs or goats, you just click on one of them, and then it'll bring you to the next page that looks like this. All right, and so with this, you click the month that you would wean the animal, and the relative weight of that animal. So you put those two in, there's a little drop down menu, and then you hit calculate. So then when you hit calculate, what it does is it gives you this chart right here. So this chart, so when I put it in, and it was for August and a 40-pound lamb, it anticipates that the lamb is going to gain 15 pounds a month. They can gain more, they can gain less. It depends on your management, but the app is built on that. So this is telling me that in September, that same lamb would be worth $23 more. For it predicts the change in the market month to month and the change in the market by weight class. But it's very important that you get this cost to gain slider accurate for your scenario. So right now I have it on 25 cents a pound of gain. This would be indicative of a lamb that was still on pasture and we're just supplementing them a little bit, but pasture conditions are good and they're growing well because our cost of gain would be fairly low. And this is telling us to keep a lamb all the way to 100 pounds and we would make an extra $63 per head. The data that goes into this is a 10-year rolling average prediction. Every year is a little bit different. We can't guarantee the markets. No one can guarantee the markets in reality. And so it's just a relative prediction. But here's another example. Same thing for goats. But in this model, it predicts goats to only gain 10 pounds a month. I put the slider bar here at the bottom to be 85 cents, which is actually what our cost of gain is in the goats behind us that Daryl is going to talk about in just a minute. But in this scenario, it's telling us that we should keep the goats all the way to 80 pounds and make an extra $35 per head. So we don't just put this together and, just say in theory you should do this. We also put it into practice to give some hard data. And so Daryl is going to talk about a study that we did here, or kind of a proof and concept study. We had our own goats and we decided to see how accurate the market or the app would predict our goats' gain.
Right, so we weaned a set of kids from a ranch in Menard County, Texas. The decision was made based on information from this app to feed these kids and attempt to add value to the kid crop. And so what you see here are actually the nanny mates to the billy kids that we wound up feeding, and we've got an analysis to share with you today. So we wanted to look at this on an enterprise level. So in other words, independent from, that kid's profitability on the nanny. And there's only three numbers you need to determine profitability when you feed animals. And it's the value that the animal had when you began the feeding trial, the value that the animal has when he comes off of feed, and then the cost to feed that animal during that feeding period. So when we look at this table here, this top set of numbers are our end values. These billy kids were weaned on May 10th. We fed 95 head. They weighed 41.8 pounds. And to capture an accurate end value, we sold some siblings from this set of billies. And so this is actual pricing from producer Cell Barn here in San Angelo. And those kids were valued. They brought $123 a head. And so that's the value we placed on the billies that we placed on feed. We sold these billies on July 21st. After death loss, we sold 91 head. They weighed 73 pounds. And they brought $229 a head at the end of the feeding period. So we added around $106 per head to feed these billies for 72 days. So a critical component in this is the cost to do so. When we talk about cost to gain, the things that we included was the feed stuff. We had hay. We gave a 44% CD&T vaccination, and we had some signs of coccidiosis. So we applied Corid in the water. But our cost to gain was 83 cents a pound. Total cost to feed a billy for 72 days in this in our situation was $25.80. So ultimately, we netted an additional $80.43 per billy that we fed. When we multiply that by the 91 head that we fed, we netted an additional $7,318 to this set of billy kids. When we take out the value of death loss, we ultimately netted $6,823. And so these billies fed really well. Health was good. You can see some production metrics here in the middle that we won't go into. We can talk about opportunity costs and depreciation. We just don't have time to do that today. So why did these billies perform so well on feed? Well, this top chart depicts, the blue and the green line depict the 5 and the 10-year historical average market price for goat kids in San Angelo. And the red line depicts how we've performed in 2020. So it's been a great year from a market standpoint. Obviously, that has a big impact on how these billies fed. Another question that we wanted to look into was, how do these different inputs impact profitability? In other words, as death loss, cost of gain, markets, as they change, how does that impact our profitability per head? So on this bottom graph, you'll see on the y-axis, profitability per head. The x-axis depicts a percentage change in each one of these inputs. And so basically the way to read this chart is the more variation we see top to bottom implies that that particular input has a bigger impact on profitability. And so based on the numbers we experienced through feeding these billies, the biggest impacts on profitability were the outprice, ADG, feed conversion, and in price. I think what was interesting to me is that although we talk about death loss and feed cost a lot, compared to these other inputs, it's not nearly as important to profitability. So with that, I think there's a couple things that the most successful operations do that I've been around, and they always seem to be diversified, and they always seem to be positioned to take advantage of the marketplace. And so I think this is one tool that we can use to position ourselves to do that.
So what was remind us what the app prediction was for those goats in May,
right?
And what was the actual for this year's performance?
So the app modeled, I think, between $14 and $15 per head. Of course, that's based off the historical average. So the market this year was just that good.
Yeah. So we were still planning on making money in the trough.
That's right.
But due to the fact that the market was still up high, it just, increased the value even more. Yeah. Okay. We got any questions coming in from the chat?
How do I determine average daily gain? How do I project the gain in my lambs and goats?
So the question was, how do we determine average daily gain?
So how it's calculated is the weight when they go on feed and the weight they come off feed divided by how many days they're on feed. These goats fed at 0.433 pounds per head per day. I don't have a lot of experience feeding goats, to be perfectly honest. I know weather has a big impact. These are like I said, the nanny mates. We'll have all the metrics from them, and hopefully we can look at profitability from a reproductive standpoint. In other words, will we add more value to this nannies by having them on a high plane of nutrition and exposing them earlier than we normally would in a range setting? But
yeah, and this, these pens are set up for sheep and goats, and our cost of gain was fairly low, but they gained at 0.4 pounds a day, which is higher than what the app was predicting. And then the feed was coming out of a local feed yard, came in at $240 a ton, something like that. So that driven that cost of gain. If you were in a pasture, you really wouldn't anticipate that rate of gain, unless just phenomenal pasture conditions. You'd probably be half that. So any other questions coming in the chat box? Okay. All right. Well, I hope our team is ready over at the forages site, and we're going to cut away now and go to Dr. Reagan Nolan over at plots where he has some summer forages to demonstrate.
We're there.
They're there?
Y'all going to give me... Hello, my name is Reagan Noland. I'm an extension agronomist here at the Texas A&M AgriLife Research and Extension Center in San Angelo. And I'm going to talk just for a few minutes about different forage options, ways that we can plant and manage forage species, different varieties, mixtures that really play into our sheep and goat production, primarily in this region of West Central Texas, but this kind of information applies across the state. Behind me here is a sorghum sedan hybrid that has been irrigated and fertilized really well. And this is a good example of the growth potential for a forage species like this in our part of the state. If you've got the means to actually manage a crop like that, These are capable of producing a lot of tonnage, being very productive, and these kind of hay grazers or sorghum sedans are a long time standard for warm season forage in this part of the world. It's really hard to beat in terms of tonnage and heat and drought tolerance when it comes to these high biomass type sorghums. Another option that we've got in another field here at the center is pearl millet, and we planted a dryland field of that in late June. No, it was early July 3rd, and that's what I'm holding here in this hand. And so this is dry land. It hasn't rained hardly since then. We had 1 or 2 rains on it to get it up and going, but we planted that in a combination with sun hemp. And so that's the other thing I've got here. Sun hemp is a tropical forage legume that has been kind of popularized recently in the United States as a cover crop or a forage crop. So legumes fix nitrogen, they're high in protein, and they make really great both cover or forage crops if you can fit them into your environment or your system. So what we've planted over there, both of these are very heat and drought tolerant. They're up, they're growing, they're not ready to graze yet. Obviously, we don't have very much going on here, but as soon as it starts raining, we've got a great opportunity to start making some forage. So we're kind of waiting for those fall rains to come in and kind of help us take off with that. When we get into managing dryland forages, especially in West Texas, it's important to consider rainfall patterns and to try to consider these types of crops that can withstand the heat and the drought that they're likely to go through. We won't be lucky enough in most years to get away with forages that might be common in the eastern U.S. So we have to select species that are going to be better for the heat and the drought, and that will take advantage of rain when we get it. So these are 2 species that represent that very well, and we're excited to see what they do. Earlier today, I ran out to some other plots we have at Wall, just east of San Angelo, and pulled some samples. This is iron and clay cowpea. It's another forage legume that we grow in some of our research plots. It hasn't been grown very widely in this part of the state, but that we're pretty excited about. There's a few different varieties of cowpea. The one that we're focusing on the most moving forward is called Ace cowpea, and that's a release from the Texas A&M AgriLife Research Program. This other one, these were growing side by side, so you can tell which one was the obvious winner in terms of tonnage. This other one's called Red Ripper, and it actually has beans on it. Cowpeas are the same as black-eyed peas. Yeah, if we can zoom in here, I'll show you. The cool thing about these Red Rippers, see the peas in there? And so, I mean, if I was a sheep, I mean, I might even eat these for lunch. Those look tasty. But the difference between these types of varieties is these have already flowered and set pods, and so they're not doing much more vegetative growth. If you want something that's going to continue to produce forage throughout the year, you might need something that's more indeterminate like this, that's just continuing to put on leaves and stems. But the reason these are called Red Rippers is kind of interesting. You see the thick taproot that they have down at the bottom. It's really great from an agronomic standpoint to have a diversity of root structures, different plant types growing in the field together, because you can take advantage of nutrients and water and improve soil structure and soil organic matter with those different roots down there. So these are some kind of exciting species that we're looking at as well, and good options to consider if you're looking for a high protein warm season forage to include with something like sorghum sedan grass or millet in a mixture. So I would certainly recommend looking into that and consult AgriLife Extension or some of our publications for a bit more detail on those. The other thing that I don't have in front of me, because here we are in August, that I'd like to talk about is our cool season legume and small grain forage crops. So we have variety trials around the state looking at winter small grains, including wheat, oats, barley, rye, triticale, and we do clippings on these throughout the winter and into the spring to see which ones really give us the best forage yield at different times and overall. The important thing to consider when you're selecting a winter small grain or even the winter legumes that I'll talk about in a bit is when you need the grazing. So we know that wheat and oats, for example, generally give us more tonnage, oats in particular in the fall. And then in the middle of the winter, you're going to get more off of wheat and oats than you might off of rye or triticale. But later in the spring, if you're cutting hay or grazing once it heads out, you're going to get a lot more tonnage off of those other species. So all of this information by harvest and across different varieties is organized in our variety testing publications. If you go to varietytesting.tamu.edu, that's the website with all the information from several years, several sites across the state. I get the question a lot from growers about, where do I get this random variety of seed that you're recommending? Because what they go buy at the feed store might just be the only wheat that they have bagged up and ready to sell. I would encourage y'all to consult ag dealers, any ag product or chemical dealer, any feed store. I've talked with a few of them to verify, and I don't want to speak locally because this is a statewide program, but consult those businesses, go to them early whenever you know what variety you want and ask them if they can get it for you. In most cases, it can be thrown on a truck somewhere and shipped or moved around. Those seeds are available. They're just not always kept locally in stock. So that's something that we kind of need to work on with which varieties we keep handy, but it's a question that we get a lot. So the last thing I want to talk about is winter legumes. So I mentioned cool season small grains. We've looked a lot at cool season legume, forage, and cover crops as well. We don't have any of these to show you again because we're here in the middle of August, but of the species that we've seen really great potential for, planted in mixtures or alone in the fall, we've got winter peas, so Austrian winter pea, or you'll just see them called winter peas, hairy or crown vetch, and then sweet clover is one that we've kind of learned about recently. We don't plant a lot of clovers the farther west we get. The drier climates and the higher pH soils generally don't fare well for clovers, but there's a variety called Silver River sweet clover that has done outstanding in our trials. We only have 1 year of data on this, so I'm going to be careful to make full-blown recommendations, but keep an eye out for some of these clovers coming out of the A&M breeding programs. They're publicly available and a really great option to look into moving forward. So I think we've got a minute or two left. Are there any questions out there that could be looped into us?
So I missed some of your other stuff. What, in a dry land or actually an irrigated situation with some of these summer forages, what's the total tonnage that a person could look at getting out of that? And what's the relative grazing value? Do you have any of those numbers?
Yeah, roughly. And tonnage, when we start talking about forage like this, needs to always be put into context of moisture. So I hear guys talk about getting 15 tons of silage off of this farm. Well, that's at 85% moisture. And that's important to realize. Most of what you're talking about is water. And so if we get a few tons of actual dry matter, which is a good way to kind of back that off, then we're still doing really well. And so that's kind of an important thing to consider. As far as nutritive value goes, you've got the brown midrib trait in a lot of sorghum sedan hybrids that reduces the lignin concentration and then increases the overall digestibility of that forage. But overall, these grasses are great for tonnage. I'm really looking to integrate these legumes where we can to boost our quality and our nutritive value because they're loaded with protein.
Awesome. So, well, thanks. You can always go to our website, sanangelo.tamu.edu. It's got links for both our research staff and our extension staff. So if you want to follow up with Reagan on some things, he's there on our website. And I guess this is the point we're going to cut away to our last session. And Mr. Jake Thorne is going to lead that session and talk about the Smart Shepherds, which is a Bluetooth technology to match parentage. So hopefully we're ready to cut away. Those guys are ready over there. And so take it away.
Hello, everybody. Jake Thorne here again. I'm an extension associate with Texas A&M AgriLife. Thanks for having me again. I had the opportunity to visit with you this morning about some genetic selection practices, particularly for parasites. And we're going to circle back to that, whether we're talking about genetic selection tools and how to integrate that into your management, or maybe you just want to know more about how your nannies and ewes perform production-wise out of the pasture. One of the key elements to that equation is knowing pedigree. And so knowing pedigree, at least knowing which lambs belong to which ewes or which kids belong to which nannies, for the majority of the time, we've been limited to just matching up kids at birth or single sire mating to get the sire side of that equation. Or if you're real handy, you know, being able to watch kids or lambs and which ewe they pair up with out in the pasture. But all of that takes a lot of labor input, time and effort. And so there's some new technology that's been developed in the last decade or so, and even within the last few years, that really makes that job of capturing pedigree a lot easier. So here at the station, we've implemented DNA. We've been using tissue and DNA to match up both sire and dam. That allows us to do things like multi-sire mate out in the pasture, and also we don't have to be present at that birth event when they do kid or lamb. But the DNA, admittedly, is a bit cost prohibitive. It's still expensive. It makes sense for us because we're trying to perform research in addition to capturing that parentage information. And maybe if you're a seed stock producer, that cost is still worthwhile. But there's still some inhibitions, or at least some concerns about how much of an expense that DNA technology might be. So some other things that you can try are actually some Bluetooth technology. So this is really neat. This has just been released within the last year or two. And this is a Smart Shepherd collar. And how this works is actually the sheep wear this collar. I'll show you here. Pretty simple to apply. Okay, and that is a Bluetooth technology. And what that does is that animal wears that collar for 48 hours. We put it on the lambs and the ewes, and we turn them back out in the pasture. And it measures proximity from one collar to another several thousand times over that 48-hour period. And then eventually we download that information, put it into a software, and it matches up which lambs belong to which ewes. So when you put that collar on, you certainly need to assign it. So that's actually what this little handheld device does. So put that collar on, you read it, you assign that animal's ear tag number. Actually, if you have an EID tag, it makes it even that much more simple. You can just scan that EID tag, it automatically assigns that collar to that animal, then turn them out in the pasture. Like I said, 48 hours later, you're able to bring that animal in and receive that information. So we had got a hold of these collars just later in the spring this year, and we wanted to try them out. And so we actually tried it out on these very Rambouillet lambs. They were a little bit older than maybe ideal. I think when we visited with the company that developed these, they state that marking or docking about a month of age is probably the prime time to try this out. These lambs were closer to that 60 to 75 day window when we tried it out, but we wanted to give it a shot. And so we put them on, we turned it out. We turned those actually out in the 16-acre pasture behind us, and we brought them in. And what we learned is we were able to match 85% of the lambs to a ewe. Now, this was our first go at it. We were still learning how to use the technology. And then, like I said, these lambs were pretty old. They were ready to come off the ewes at the time. And so we still think that 85%, knowing which of those lambs matched to a ewe, was pretty good. We also tried it again, a second time, with some meat goats. And this was a little bit different story. Again, the kids were a little bit older than ideal, and they were ready to be weaned, but we tried it, and we were able to match over 95% of those kids to a nanny. But what was really interesting is when we put those collars on, we went ahead and recorded which nanny still had a bag. And what we found was a pretty high percentage of those nannies, like 20%, that had a bag and that obviously had a kid, did not match to a kid that existed. And so what that really told us is potentially there was some predation issues, or maybe those nannies lost a kid for some other reason, or mismothering. And so really, all in all, this arms us with a lot more information about how productive our females have been during that kid or lamb rearing process. So I think that that's really interesting. Again, that provides us just with a bit more information about capturing efficiency and data for recording which animals are superior for those types of traits. Really neat technology. We're looking forward to trying this out again later this fall. Again, we're going to try it on some younger lambs and kids this time. We're also going to have DNA information to really compare that to which lambs really truly belong to which ewes. So I think that that's really interesting. If there's any questions, I'd certainly be willing to entertain those at this time.
Yeah, Jake, so one of the questions is, you talk about
Sure, absolutely. So the question was, how much does this cost? And that's a little bit tricky to understand. First of all, this is a tool that you're going to use maybe once or twice a year. And so the model that's been developed in some other countries, mainly Australia, somebody that, a consultant that owns these collars. Now, you call that consultant and you request their services, and they bring the collars out, they assign the collars to the animal, and then they capture that information and share it to you. And they do that for just a few dollars a head. Now, that's not something that you can currently do in the U.S., but again, we've been in contact with the company, we've been working with them. I know they're really interested in getting this product placed in the U.S., and so that's probably a model that they want to follow. Again, we want to get down to where it's just a couple dollars a head to learn this information. I think that makes a lot of sense for seed stock producers, but also commercial producers that want to know the productivity and efficiency of their use. So again, I know that's not a real straightforward answer as exactly what the cost is. The units are probably much more expensive than that. But again, I don't necessarily want to encourage you to just go out and buy them. I think that if we, purchase that service or enlist that service from somebody, it's going to be a lot cheaper. Okay.
Okay, so that concludes the technical part of this morning's presentations. We're going to go to Reed Redden and Robert Fritz, and they'll present some concluding remarks.
All right, welcome back.
Hold.
Okay, welcome back. This kind of concludes our field day session. I think it's been very successful. I was extremely nervous about all this technology and things not working, but for the most part, I think things have gone well. We hope you all have enjoyed getting an update of the things that are going on here at the Research Center. If you have ideas or suggestions, you can always email us. I encourage you to go to our website, sanangelo.tamu.edu. You can keep up with us, find contact information, follow our Facebook page, Texas A&M Sheep and Goat Extension. But we're not done. We're just taking a lunch break. Robert, you want to tell our audience what to expect this afternoon?
Absolutely. Great field day, Reed. Really glad the technology is working out for us. We're excited. Like I said, it doesn't end now. We're going to take a short lunch break, let everybody go grab something to eat. We're going to start back up with the Texas Sheep and Goat Expo at 1 p.m. We're going to move it indoors this afternoon. We're going to try to get out of this 109-degree heat this afternoon that we've got here in San Angelo expected today. So I want everybody to go take a break, get some lunch. We've got an exciting cast of speakers coming this afternoon and tomorrow as well. But this afternoon, we're going to be looking at some direct marketing panel. We're also going to have a very unique mock auction that we're thinking everybody's going to be wanting to come back and see. We've got several topics, and then we're going to finish off the day this afternoon, Reed, with an international viewpoint. So we've still got a lot left today. Hope you'll come back and be with us, but we're going to take a short break and see you back at 1 p.m.
All right, if people log off, they come back into the same link as what they used this morning, correct?
Absolutely. It's going to be one link all day long. So come and go as you need, but we'll be here. And like I said, make sure to be back with us at 1 p.m.
Yeah, we apologize for those that were trying to watch Facebook Live. We've had some technical issues there, but we're at that link. And so the second link is just for tomorrow.
Yes.
Come back to the same link. We'll see you after lunch.
See you then.